Welcome, one and all. It's nice to see so many familiar faces and lovely to meet new people that I haven't yet met. My name is Mervan Singh. Most people know me as Sunny, so please feel free to call me that. I'm the Vice Dean of Research here at Loyola University Stritch School of Medicine. Excuse me. And it's my pleasure to welcome you to our Health Sciences campus. And in partnership with Cognition Therapeutics, Susan Catalano and team, it's really our pleasure to have the opportunity to host you all on our campus. This is the CTRE building, the Center for Translational Research and Education. It's our newest building. As some of you may already detect, it still has the new car smell, so to speak. But if some of you are interested in seeing sort of the layout of the building, I'd be happy to sort of take you for a short and quick tour. Again, I want to extend a tremendous sense of gratitude to Susan, Aline, and her team from Cognition Therapeutics for really doing the heavy lifting in bringing us together again for the fourth time. An important topic for sure, an emerging topic in some circles, but nonetheless something important for a variety of different fields, as I'm sure we'll all learn today. So a couple of housekeeping matters. Restrooms. If you go out these doors, and as you see the security guard right in front of you as you walk out these doors, just go past the security guard, and on your left will be the restrooms. If there's any sort of AV-related issues for any of the speakers, we'll try to get those addressed as quickly as possible. But otherwise, I look forward to some great discussion on a very exciting topic. And without any further ado, let me introduce to you someone who doesn't need any introduction to this group, Susan Catalano.
Okay. Alrighty, so thank you so very much, Sunny. We really, really appreciate it. This is you and your executive assistant, Maria, and the events manager, Nancy, really were wonderful in helping us host this event here. So as Sunny said, I'm Susan Catalano. I'm the founder and chief science officer of Cognition Therapeutics. And I wanted to certainly start by thanking the Society for authorizing this satellite symposium and including us in our materials. And I want to thank my colleagues for traveling here and getting here early so that you could participate in this symposia. Certainly, we have to thank Aileen Sherwood, who is out of the room right now getting water for the speakers, but for really just doing an enormous amount. Oh, there you are. Okay, Aileen, please wave. Thank you, Aileen. Yeah, without her, it just simply wouldn't have happened. As well as Carrie Moore, Kelsey Mazzoni. Where are you, Kelsey? Wave. There's Kelsey. Nick Izzo. It's right here. And of course, our CEO, Ken Mock. So he's right there. Yeah. So we're going to plan to publish a short meeting proceedings article in the Society's journal, and we're going to be helped in that endeavor by Hillary Shaler. So Hillary is right there. Wave, Hillary, so everyone can see. And so she might be sending you drafts for comment down the road. And so want to also thank all of the colleagues who'll be speaking here. This is really going to be an exciting session. So the last time that we got together was last year at the 3rd Nano Symposium, and the publications in the field continue to accelerate, so there's lots to talk about this time. So I'd like to start, like I did in the previous sessions, by giving a very brief overview for folks who might be unfamiliar with the field, and then introduce the first speaker, who is Dr. Wayne Bowen. So I want to apologize in advance to any of the colleagues whose work I will not have the opportunity to cite appropriately in this presentation. So here we go. So the history of Sigma receptors goes back to the initial studies on SKF 10047. It's an opiate-like compound, and it had profound CNS activities. And Gilbert and his colleagues found that those activities and the binding of the drug were not blocked by opiate antagonists, and so they proposed the existence of a new receptor they called Sigma, which was named after the compound itself. Early studies focused on the psychotomimetic effects of this compound, and that was later found to be a result of the drug's interaction with PCP receptors. And we now recognize, of course, that Sigma-2 receptors do not play a role in opiate pharmacology. This was followed by some very seminal work that was done by Wayne and his colleagues to define further subtypes of this receptor based on the differential binding of the ligand we all know and love, DTG. So in the presence of saturating pentazocine, which blocks the sigma-1 site, which despite the name is a totally unrelated protein, DTG binds preferentially to sigma-2 sites, which are in relatively low abundance in the brain, but in high abundance in tumor cells, such as PC12 cells. And displacement of radiolabeled DTG in the presence of saturating pentazocine is still the canonical definition of a sigma-2 ligand to this day. So Sigma receptors have continued to be of interest to CNS drug development because of the effects in schizophrenia, depression, anxiety, pain, cocaine withdrawal, anti-amnesia, neuroprotection, but there have been no clinical successes of selective Sigma-2 ligands as yet. So in the absence of an identified protein corresponding to the Sigma-2 receptor, the one thing that most folks agreed on is that some Sigma-2 ligands caused an increase in cytosolic calcium and a resulting caspase activation and neurotoxicity at high concentrations. But they also play a role in cellular metabolism, and these studies were pioneered by Wayne Bowen and his colleagues, and we'll be hearing more about this today from Wayne in his talk. So all of this changed when Bob Mack published that seminal 2011 study identifying the PGRMC1 protein complex as the Sigma-2 receptor, which propelled the field forward in a very big way. Not much was known or published about this protein and the family members at the time, and the vast majority of it was in cancer biology and progesterone biology and reproductive tissues. But we've literally seen the field explode with more than 500 references on PGRMC1 or related topics, and the vast majority of this was in the last 5 years alone. I think our symposia can take partial credit for this, so we can all pat ourselves on the back for that. So PGRMC1 stands for progesterone receptor membrane component 1, and it's one of a series of proteins referred to as the MAPR family, or the membrane-associated progesterone receptor family. The family members are shown here. They're characterized by a single transmembrane sequence and a heme-binding domain with homology to the cytochrome B5 domain. They also have an awful lot of consensus domains that are involved in a variety of signaling pathways. This was outlined by Mike Cahill in his seminal 2007 review of the protein. And we'll be hearing an update from Mike later on today on the many additional roles in biology that this protein family plays. In 2014, we demonstrated that modest reduction of the PGRMC1 protein, only about 30%, almost completely eliminates A-beta oligomer binding to synaptic sites on neurons. And this is a phenomenon that's also seen by treating neurons with our Sigma-2 antagonist. And we propose that this Sigma-2 receptor complex modulates the oligomer receptor complex and represents a therapeutic approach to Alzheimer's disease. Since then, we've published results from our first-in-man clinical studies of Cognition's drug CT1812. It's also known as Elata. And we demonstrated that therapeutic doses of that drug have a favorable safety profile in healthy young and elderly volunteers. By way of a hint, stopping A-beta oligomer binding has a positive effect on synapses in the brain of the 80 patients that we've tested it in because we see a reduction of synaptic protein debris as well as phospho-tau in the CNS of 80 patients treated with 1812. So I'll be giving you an update about our first inpatient clinical trial treating mild to moderate patients in my talk, and Nick Izzo is going to talk more about the signaling pathways linked to our drug in 80 patients in his talk. Now, not too many folks believe that PGRMC1 directly binds progesterone itself, but Sunny Singh and his colleagues have demonstrated that it does mediate P4-evoked BDNF release from glia in the brain, and this mechanism of neuroprotection against stroke it can be inhibited by LET7I, and we'll hear more about this from Sunny in his talk. A recent paper reported another potential neuroprotective mechanism that's independent of progesterone signaling in immortalized neuronal cell lines, and there it blocks the induction of gene expression downstream of TNF-alpha. Rolf Craven, who unfortunately could not be here today, and others have published on the function of PGRMC1 mediating the process of autophagy by directly binding to MAP1 light chain 3 and UV-RAG, and certainly direct binding to cytochrome P450 and its role in steroid metabolism and catabolism, and its function to stabilize the plasma membrane localization of many different receptors, and that includes EGF receptor, NPR alpha, GLP-1R, insulin receptor, probably others as well. A major advancement came in 2016 when Makoto Suimatsu and his colleagues published the crystal structure of PGRMC1 homodimers. And these are held together by stacking of the heme groups. This is a first in eukaryotic biology. The dimer configurations required for PGRMC1 to stabilize EGF receptor in the plasma membrane and perhaps other functions as well. And it's destabilized by carbon monoxide binding and perhaps other diatomic gases like nitric oxide, which plays a role in synaptic plasticity. So PGRMC1 also interacts with INSYG and SCAP. It signals low cholesterol in the cell, and it initiates transcription of the SRI-related genes, including HMG-CoA. So it also interacts with UNC40-DCC. This is a system that's active in axon guidance, which attracted my colleague Tim Kennedy to our field. Tim's been studying the role of netrin in activity-dependent synaptic plasticity, not only in neuronal development, but also underlying learning and memory in the adult nervous system. And so we can expect to hear more on that topic that's certainly central to Alzheimer's disease biology etiology in the future. But all was not simple. So as we noted in 2016, several labs, including Carmen Abate and Arnie Rojo, published work showing that Sigma-2 ligand binding persisted in the absence of the protein PGRMC1, at least in tumor cell lines. So we ended our symposia at the time with the conclusion that not only did we have a lot more work to do to link the biology of PGRMC1 with Sigma-2 pharmacology, but that there may in fact be additional proteins involved in constituting the canonical Sigma-2 receptor, or perhaps subtypes of Sigma-2 receptor, or even a Sigma-3 receptor. Then along came this 2017 publication by Jim San, Steve Martin, and Andrew Cruz, and it said that another little-known protein called TMEM97 or MAC30 was responsible for Sigma-2 ligand binding, again in tumor cell systems. So we know that this protein binds to NPC. This is the protein that's mutated in Niemann-Pick's disease, which is a lysosomal storage disorder, and that it contains motifs that play a role in cholesterol homeostasis, but beyond that, not much is known. And then last year, my co-chair, Bob Mack, and today's speaker, Aladdin Riyad, published an important paper describing the requirement for both PGRMC1 and TMEM97 in internalizing ligand-bound LDL receptor. So together with additional work from Bob's lab, this confirms a central role for PGRMC1 in Sigma-2 receptor function in at least some signaling contexts, making it likely that Sigma-2 is probably not a single protein, but a multi-protein complex, which comes as no surprise to anyone who is familiar with synaptic biology, certainly. It was exciting for us because the closely related receptor LRP is one of the mechanisms for ApoE-based A-beta protein clearance from the brain to the plasma. And it also binds directly to oligomer receptor complex protein, cellular prion protein. So we'll hear an update on this exciting work from Aladdin later on today, and you'll hear more about the interaction of these two proteins, certainly in neurons, in Nick's talk and in Kelsey Sadleck's poster from our lab. A number of labs continue to explore Sigma-2 ligands for therapeutic indications like cancer, and we'll be hearing more about that because we're very fortunate that Dr. Carmen Abate has joined us from Bari, Italy, to update us on her work. The ligands are also very important tools to understand the pharmacology of the receptor in different signaling contexts, which is especially important in neurobiology, where state-dependent signaling and the temporal history of signaling at a given synapse is really plays a major role in brain function, in health and in disease. And we'll be hearing more on that from many of the speakers today. Many of the labs of the speakers are developing new Sigma-2 ligands. This includes the lab of Dr. Chris McCurdy and Ray Matsumoto, who have collaborated to study the role of Sigma-2 receptors in addiction. Additional references from select recent papers can be found in the handout, and apologies to those authors whose work was not included. We have a very full agenda today with a truly international roster of speakers who I am pleased to say are also very treasured colleagues and friends. So I'm really looking forward to hearing everybody's latest. Both Marti Colum-Cadena from the lab of Dr. Tara Spires-Jones, whom you heard from last year, and Mike Cahill are going to be speaking via video feed. And Bob will provide a few closing remarks, followed by a reception in the auditorium lobby right out here where the coffee is. So we're going to break for lunch in the nearby alumni lounge, and I think there are going to be signs directing you there. So we're looking forward to that too, courtesy of Sunny. So we are still at the beginning of understanding the precise relationship between Sigma-2 receptors and the functional effects of these ligands and pharmacology, and understanding PTRMC1 and TMEM97 protein functions. Bob's given us a great start at this. As you can see, we're starting to fill in the areas of this Venn diagram that are really important to understand. There's only one known small molecule inhibitor of the PTRMC1 protein, that's AG205. It's really less than ideal as a small molecule to probe the functions of this biology. So we're hoping to improve on that situation. PTRMC1 and TMEM97 biology really need to be explored with selective, high-affinity Sigma-2 ligands. And certainly the biology of the other MAPR family members needs further explanation. Signal transduction pathways downstream of each other, both of those proteins need further definition. The rich interface between the pharmacology and the biology that's central to CNS function in health and disease is really worthy of what Suzanne Pedincheska, the NIA program officer, calls a reverse translation approach. This is bringing experts in the field together and providing many opportunities for new investigators to launch their careers, and we hope a lot of them are here in the audience today. So with that, I will ask Bob to help keep us on time today and introduce our first speaker, who is Dr. Wayne Bowen. Did you want to say a few words? Okay, great, thanks. Okay, Wayne.
Can I use the lapel mic? Great. Thank you. Well, good morning. I am really excited to kick off what promises to be again this year, strangled up here, again this year, a very exciting symposium. We're in store for some really interesting and exciting work today. So what I'd like to do is is fill you in on, give you a brief update on some of the work that we've been doing with respect to a relatively novel potential function for Sigma-2, and that is the regulation of metabolism in cells. Can everybody see that? It's a little bit difficult to see. Okay, so as you know, we've been working with Chris McCurdy's group in developing some novel ligands based on this compound, SN79, which is a putative Sigma-2 receptor antagonist. As you can see, the binding data is here. It's not terribly selective for Sigma-2. And we initially set out to try and improve the selectivity of this compound in the hopes of developing a more selective Sigma-2 receptor antagonist. And in the process of working with this group of compounds, we came across this ligand here, CM764, which has an amino substitution on the phenylfluoral ring. And you see we got a little bit improvement in the Sigma-2 selectivity, but that wasn't the most interesting thing about this compound. The most interesting thing about this compound is that it turns out to stimulate some of the hallmarks of glycolytic metabolism. Now, most of the compounds that we had been working with turn out to activate apoptotic pathways in cells. But this one turned out to be different, and we were sort of surprised about this. We measure cell viability by the MTT assay, which just is a general measure of cellular metabolism. It's basically used to determine the number of viable cells you have in your well. And what Hillary observed, and this is work from Hillary Nicholson, what she observed is that there's a dose-dependent increase in MTT reduction. And this occurs without an increase in cell proliferation. So there's no change in the number of cells, even over a period of 4 days. And so what we were actually just measuring is an increase in MTT reduction. And noticing that that's a fairly strange result, I always tell my students, first assume that it is an artifact and then try to prove that it's not an artifact. And so what she did was able to show that this effect is blocked by a number of sigma receptor antagonists. So here you have stimulation of metabolism by CM764 alone. And all of these compounds are able to block this stimulation of metabolism without themselves causing a stimulation of metabolism. And you see here a number of antagonists, BD1047, NE100, BD1063. And SN79 itself is unable to stimulate metabolism, but is able to block the stimulation of metabolism by CM764. This compound, CM572, at higher doses is cytotoxic, but it is a highly selective Sigma-2 receptor ligand, and which we found that in low doses, this compound binds irreversibly to the receptors and it acts as an antagonist. So we characterize it as a partial agonist. This compound produces a calcium signal that is a transient calcium signal in the cytosol. But we found that it increases the level of ATP in cells. It dramatically reduces basal ROS levels in cells. It's much more effective than alpha tocopherol, in fact. So it's reducing basal ROS levels. But this was the most exciting finding in that there's a time-dependent increase in the stabilization of HIF-1 alpha. And as you know, HIF-1 alpha is sort of a global regulator of metabolism in cells, particularly in cancer cells. And this ligand is able to produce a time-dependent stabilization. And as you know, work on HIF-1 alpha was awarded the Nobel Prize a couple weeks ago. And Bill Kalin was one of the recipients, and Hillary is now working in his lab. Very exciting for Hillary. So Hillary went on to characterize other members of this family, and when we started looking at a number of analogs of SN79, what we found was that, and what we did was we varied the X substituent in the heterocycle and the substituent at the 6th position here. And what we found was that when R is isothiocyanate, regardless of what the heterocycle is, that these compounds all bind irreversibly to the receptor. And what we incorporated the isothiocyanate in hopes of achieving selective irreversible binding to the Sigma-2 receptor, and that is what we found in her initial paper, CM572, is an irreversible, highly selective binder to the Sigma-2 receptor. And all of those compounds with isothiocyanate induce programmed cell death. If the R substituent was anything else, either nitro, amino, methyl ketone, or fluoro, regardless of the heterocycle, these compounds, most of them stimulated metabolism. So there was this divergence in the effect of these ligands. And so what we wanted to do is further characterize the pharmacology of this effect using additional ligands that came out of this study. And the ligands, the additional ligands were CM571 and WA504, structures shown here. And these are their Ki values. They have good affinity at Sigma-2, but they're not selective for Sigma-2. So these compounds all stimulate MTT reduction, so they're all consistent with what we had seen with CM764. And it's shown you earlier that these compounds, that this effect is blocked by a number of Sigma antagonists generally, and then this highly selective Sigma-2 antagonist. But we wanted to look at some additional compounds in this series just to ask some other questions. So this is a related compound, structurally related compound that lacks good affinity at either Sigma-2 or Sigma-1, and these binding values are as defined by tritiated DTG binding. You can see that this compound, which lacks affinity at either receptor, fails to stimulate MTT reduction. Another thing we wanted to look at is these compounds are not very selective for Sigma-2. They have significant Sigma-1 affinity, as you can see here. So we wanted to know, well, what's the possible role of Sigma-1 in this? Now, we knew that plus pentazocine, which is a Sigma-1 agonist, does not stimulate MTT reduction. And Sigma-2 antagonists, Sigma-1 antagonists like NE100 and BD1047 also don't stimulate metabolism. But we wanted to look at this a little further. As you notice here, these antagonists are usually considered to be Sigma-1 antagonists. They lack terrible selectivity for Sigma-1. They turn out also to be Sigma-2 antagonists, but we wanted to look at this a little bit further. This compound here sort of told us that it is selective for Sigma-2, but we wanted to look a little more closely. So here you have a compound in this series which has reasonable Sigma-2 but lacks good Sigma-1. And this compound retains its ability to stimulate MTT reduction. So that's consistent with Sigma-1 receptors not being involved. And another thing we did was we took fragments of active compounds. So the active compounds are basically composed of these two fragments linked by a 4-carbon linker. And neither fragment has Sigma-2 affinity, and this fragment has some weak Sigma-1 affinity, but the heterocyclic fragment has no Sigma-1 affinity, and neither of these compounds is able to stimulate MTT reduction. So that's all consistent with the need to have Sigma-2 activity. So in terms of the pharmacology, using additional compounds, this is all consistent with being mediated by Sigma-2 receptor pharmacology, at least as defined by competition with tritiated DTG. There's still the huge discrepancy between the potencies of these compounds and the binding affinities, which are in the nanomolar range, and then the potencies are all in the micromolar range, which could indicate that there are other proteins being involved, whose pharmacology is simply predictive by the ability to displace DTG. And I'll say a little bit more about that later. Now, another thing we wanted to look into, we wanted to sort of look at the mechanism. And we haven't gone deeply into this, but one of the first things that we did was we wanted to ask, what's the time course of the development of this metabolic effect? And so here what you see is CM764 treating SK-NSH cells for various time points to determine when this effect shows up. And you see here that with 1 hour of treatment with CM764, there's no ability to stimulate MTT reduction. If you go to 3 hours, you start to see some reduction, some stimulation of reduction, but it's very variable and not very robust. By 6 hours, you now have a robust and stable ability of the compound to stimulate MTT reduction, and that is almost maximal because we don't see much more if we go out to 24 hours of treatment. So there's a time dependence in the development of this effect. And that time dependence actually correlates somewhat with the time course for the development of HIF-1 alpha stabilization. So Hillary showed that you see a little bit of HIF stabilization at 6. By 12, you have considerably more, and it's pretty stable by 24 hours. So somewhere between 6 and 12 hours, you have this stabilization of HIF. And then it corresponds with later, by 24 hours, the production of VEGF. And VEGF is a target gene for HIF-1 alpha, and VEGF is an angiogenic factor. The other thing we wanted to investigate was the calcium signal, because all of these compounds produce a calcium signal, and we had for a long time known that the calcium signal is not part of the mechanism for cytotoxicity. And so here what we see is that, looking at CM764 and 571, there's a dose-dependent release of calcium. These are Fura-2-loaded cells. Looking at cytosolic calcium, that's 10 micromolar and 30 micromolar of the compound. But what this is showing additionally is that if you pre-treat the cells with dapsogardin to empty the ER stores and then stimulate the cells with the CM764, you completely eliminate that calcium signal. So that calcium is coming from the ER. We don't know what the mechanism is. It doesn't appear to involve IP3, but we're in the process of trying to figure out where this calcium, how this calcium is being released from the ER. And this is just showing the WA504 also is able to release calcium from the ER. Now, what is the role of this calcium? Well, there's literature saying that HIF-1 alpha stabilization is related to these types of calcium signals. So both the calcium signals being involved in the initiation of HIF-1 alpha stabilization, as well as HIF-1 alpha being involved in regulating those proteins that cause cellular handling of calcium. So it could be that HIF is playing a central role in, that this calcium signal is somehow playing a central role in the ability of HIF to become stabilized, and then that downstream is leading to this metabolic effect. So does any old calcium signal then cause the stimulation of MTT reduction? So these cells express muscarinic cholinergic receptors. Carbachol gives a very robust calcium signal in these cells, but carbachol fails to stimulate MTT reduction. So you need to have a molecule that's binding to the sigma receptor in order to get this stimulation of MTT reduction if the calcium signal is in fact related to HIF stabilization and this metabolic effect. You need to have sigma binding affinity. Now, we wanted to look at whether Sigma-2 TMM97 is involved, and we're in the process of CRISPRing out TMM97 and PGR-MC1 in these cells. We don't have that data now, so maybe next year I'll be able to present to you what's happening with that. But there is some interesting data that surrounds TMM97. Now, if you remember, Hillary had shown that these compounds that bind irreversibly to the receptor that has this isothiocyanate group cause programmed cell death, and then these other substituents produce compounds that have this metabolic stimulatory effect. And last year at this symposium, I talked about the comparison of these monovalent and bivalent Sigma-2 ligands. So here you have CM571 and CM572, which is the isothiocyanate compound that Hillary showed is cytotoxic. And I talked last time about this compound, MAM03055A, which is really a dimer, if you will, of CM571. And we compared the ability of these cells to affect cell viability. You can notice that MAM is very selective for Sigma-2 over Sigma-1, so it's about 60-fold selective. And the surprise was that MAM, as opposed to being similar to 571, is actually more similar to CM572. So MAM is cytotoxic, and this decrease in MTT reduction is actually cell death. MAM induces an apoptotic cell death with bid cleavage and other downstream markers that we had observed with other cytotoxic Sigma-2 ligands. And the other surprise was that MAM, like CM572, doesn't come off of the receptor. It binds pseudo-irreversibly. Now, there's not a moiety in the molecule that can react covalently with the receptor like there is with the isothiocyanate. But for some reason, it does not wash out of the receptor very readily. It may have a very, very slow dissociation rate. But CM571 binds reversibly to the receptor and stimulates metabolism. So these two irreversibly binding ligands are cytotoxic, and this one is metabolic stimulative. And when we look at what these compounds do to TMEM97, what we find is that CM571 has no effect on the levels of TMEM97. This is just treating the cells 30 micromolar for these various time periods, and then measuring TMEM97 in a cell extract by Western blotting. But MAM produced this time-dependent loss of TMEM97. So this irreversibly or pseudo-irreversibly binding ligand causes this time-dependent loss of TMEM97. Now this may be related to the data from Bob's lab showing that you'll hear more about later, that there's an internalization of TMEM97 along with PGR-MC1 and the LDL receptor. We don't really know what this means, but it could be explaining or giving some explanation for why these two classes of ligands may be different. So we have this one is cytotoxic, this one is metabolic stimulative. We don't know what the relationship is, and it's going to require further study. So in summary, the metabolic stimulative effect of SN79 analogs has clear Sigma-2 receptor-like profile, with the exception of the discrepancy in binding affinity, at least as characterized with DTG. The time course for the development of the metabolic effect is consistent with the time course for stabilization of HIF-1, and the Sigma-2 ligands that are metabolic stimulators induce a transient calcium signal from stores in the ER. And those types of signals have been linked to HIF expression, and the non-sigma calcium releaser failed to produce the metabolic stimulation. Now, HIF is involved in the adaptation of cells to hypoxia. So HIF affects angiogenesis and blood supply. It also affects glycolytic metabolism. And Sigma-2 receptor and HIF could be potential regulators of the Warburg effect, which is how cancer cells have adapted to living in hypoxic environments. So the Sigma-2 receptor may cause an increase in calcium, an induction of HIF by some mechanism, and we've shown that upregulates VEGF, which gets you over here to increasing blood supply. We're now in the process of getting more concrete readouts for metabolism other than just looking at MTT reduction. So we're doing some seahorse experiments. Hopefully I'll be able to talk about those next time. But the preliminary data that we have does suggest that there's an increase in lactate extrusion from the cells with measuring the ECAR and the seahorse apparatus. And hopefully I'll be able to talk to you about that more next time. So the possible implications are, in cancer, this is consistent with upregulation. It could be involved in Warburg glycolysis and enhancing tumor angiogenesis via these VEGF secretion. And it'd be interesting to see what this effect looks like in neurons, which could be production from oxidative stress with that reduction in ROS levels, protection from hypoxic stress by the upregulation of HIF, and this could have implications for these types of compounds in stroke and neurodegeneration. The divergent effects of these analogs could involve effects on TMEM97 levels. As I showed you, the irreversibly binding analogs induce cell death and cause a loss of TMEM97. The reversibly binding metabolic inhibitors have no effect on TMEM97 levels. Alternatively, these effects could be mediated by different classes of Sigma-2 like proteins or different binding partners such as PGRMC1. And what we really need are better radioligands so that we can fish out these two effects. It could be that what's happening is that DTG binding, which is a good ligand for TMEM97, may simply be predicting the pharmacology of related proteins that don't bind DTG. And so we could be seeing overlapping pharmacology with these compounds acting through compounds that just don't bind DTG, but they have such a similar pharmacological pattern that they look like they're Sigma-2 mediated effects. That's just a hypothesis, and we need more work to figure that one out. So acknowledgements. So most of the work that I showed you was by Bridget McVeigh, who was a master's student in our lab. Sherry Liu did some of the work, and then Hillary's work was also cited. And then these are our chemistry collaborators, Chris McCurdy, Christophe Massanju, and Marco Martinelli, and then this is the funding. Okay. Take questions.
Very interesting work. I wanted to ask you, and I think you kind of were addressing it, that you're going to take this into the seahorse to see whether or not you look at mitochondrial function. I would maybe like to put in a plug to maybe look at mitochondrial fission-fusion dynamics.
Sure.
The reason is some years ago, I think thapsigargin's been shown to actually influence mitochondrial fragmentation, and that could potentially influence the data, to say nothing of the fact that MTT reduction is a product of mitochondrial activity. So there may be an opportunity to perhaps distinguish between the endoplasmic reticulum as the source of calcium versus the mitochondria.
Versus the mitochondria, right.
But it just struck me as that, and I thought I'd share that.
Right. So my understanding is that MTT reduction occurs both in the cytosol and in the mitochondria by NADPH and NADH-dependent reductases. And it's interesting that the compounds in this series and other series that we've looked at, actually the cytotoxic compounds, do so by causing mitochondrial depolarization. So all of this could have something to do with regulating how mitochondria function. I'll give you a look at it. And we do have the preliminary data that we have with the seahorse does suggest that there are alterations in ECAR, but we don't have, I didn't want to show that data because we haven't yet optimized the conditions for that, but hopefully next time we'll be able to talk about that.
Thanks, Wayne. I'm wondering if some of your compounds that are acting through different mechanisms there, can you tease apart the calcium and the mitochondrial effects on HIF? So I'm just wondering if some of the compounds that are stimulating mitochondrial activity are actually causing a very localized hypoxia, and that's leading to the HIF, or if there's some more direct activity.
It's possible because we see that dramatic drop in ROS, right? So there could be sort of a feedback mechanism that is related to that. Where calcium comes in, calcium has been shown to be upstream of HIF and also downstream of HIF. So HIF can regulate calcium dynamics as well. But yeah, any of those mechanisms might be coming into play.
Okay, last question. So you mentioned the time-dependent loss of TMM97 that could potentially be due to internalization. Did you have
we
any?
don't have any evidence that just feeding on what has come out of Bob's lab
Right.
with the internalization of the complex. This could be what we're seeing. I don't know if we're going to hear more about what happens to the TMM after it gets internalized. Does it get degraded or does it come back to the cell surface? We don't know. But this data was just total cell extract, right, from cells that had been treated. So I don't know whether that's degradation or
Right.
or turnover, increase of turnover. We don't know yet.
All righty. Okay, I think we need to move on to our next speaker. So our next speaker is attending via video feed, and this is Marti Colomcadina from Dr. Tara Spires-Jones' lab. And there he is. Wave, Marti. Can you hear us? All right.
Yeah, I can hear you. Can you hear me?
We can, and the sound quality is excellent. So, so Aileen is putting your slides up. Just give us a second. Let's go full screen.
Ryan's coming down. Oh, there we go. Okay, Marty, you're up.
Okay. Thank you very much. Can you see the cursor also?
No, right now I don't see your slides.
No.
Can you hit share on your end?
I'll share
Oh.
the screen. You should be able to see like a normal conversation. Do you want me to do it again?
No, we don't see his video.
Can you see the power chain?
Uh-huh.
All right, I think we're all set.
Yeah?
Thanks, Marty.
Okay, thank you very much. So, wait a second. Should put the video. Okay, sorry for this delay. Good morning. I will present the results from a study that we are doing where we are investigating the synaptic localization of TMM97 and the potential interaction with beta-amyloid in human Alzheimer's disease cases. I will briefly explain why and how we do that, and I will show some of the results that are still preliminary because this is a study that is ongoing. So as an introduction, the neuropathological hallmarks of Alzheimer's disease is this aggregation of beta-amyloid extracellularly in form of this plaque that you can see in this picture, or the intracellular aggregation of phosphorylated tau in form of neurofibrillary tangles, neuropil threads, and both aggregates we know that they follow a hierarchical pattern, a sequential hierarchical pattern through different brain regions, and it correlates more or less with some of the symptoms of the disease. Although that is not clear, the relation of these peak aggregates with the toxicity, with the massive neurodegeneration that we see in Alzheimer's disease. And in fact, evidences from in vitro and animal model studies suggest that probably are not these big aggregates, but smaller forms or oligomeric forms, the ones that may be closer related to the neurodegeneration. So we have this on one side, and in the other side we have massive synapse loss. And actually, the synapse loss is thought to be one of the earliest events in Alzheimer's disease pathogenesis, and is in fact the best correlate of cognitive decline, as was shown in, for example, this study of 2006, where you can see that the number of synapses is lower in those cases that have a lower cognitive rate. So we have on one side these small aggregates that may be potentially toxic, and we have the synapse loss that we know that is an early event and that correlates very good with the cognitive decline that we see in Alzheimer's disease. And then it seems logical to study them together and see if we can understand the pathology with the synapse loss. The problem here with human brain is the size of the synapses. So with electron microscopy, you can see here nicely a presynaptic terminal and a postsynaptic terminal. But if we want to label also our pathological protein, we will have some problems with light microscopy. So let's say that we want to label the postsynaptic terminal that we see in this diagram as red, the presynaptic terminal in green, and our protein of the study, in this case, maybe TMM97 or beta-amyloid. So with a confocal microscope, for example, in the lateral plane, in the XY plane, we will have enough resolution probably to resolve a single synapse terminal, but especially in the axial plane, we will see something like that. So we will see an overlay of those channels, and we will not be able to resolve single synapses and to locate our protein of interest. So the approach that we are using is called array tomography, and is this 70 nanometers consecutive sections. So we will end up having something like you can see here, where we have in each consecutive section a part of the synaptic terminal, and we will be able to resolve both the synaptic terminals and our protein. In the lab, to do this, we collect fresh brain tissue that is embedded in an acrylic resin. Then we produce these 70 nanometers each sections that you can see here, and around 20, 30 consecutive sections. And this first part is similar to an electron microscopy procedure, but from here on is normally immunofluorescent, and you can image either with confocal or fluorescent microscope. So after this, we will end up having something like that. What we have in this example, the postsynaptic terminals, presynaptic terminals, and nucleus. And those images needs to be processed with alignment, segmentation. Here we see an example of that in a single 70 nanometer section with in blue the postsynaptic and red the presynaptic terminals. And we will end up having something like that. That is a three-dimensional space where we will have those objects, those synapses, and our protein of interest. We are doing this with an in-house algorithm that you can find in this GitHub here. So already in the first study that was done with human AD cases, that was already led by Prof. Terence Parsons, what they saw was that especially in the vicinity of the beta-amyloid plaque that here we see in blue, there is a synapse loss. So for example, here we can see the quantification of the PS95 density, and especially close to the plaques, there is this synapse loss. But even more, what was interesting is that in some of the synapses that especially close to the plaque, we can see beta-amyloid. So here we have the first demonstration that have been replicated many times that beta-amyloid can be found in synapses of Alzheimer's disease cases, especially in the vicinity of the plaques. So now the next step is to think if it's binding to some protein and if there is this potential toxicity also. So here is where it comes TMM97. We found, as you well know, it was recently identified as the Sigma-2 receptor, and there are many compounds that selectively binds to Sigma-2, and that it has been shown that it reduces identification. So it's a good target to investigate this potential binding. And also recently in a study that we did and that is still under revision, we compared by proteomics synaptoneurosomes from AD cases and from controls. And among other results, we found that TMM97 was increased by more than 2-fold in AD cases when compared to controls. So that's why in this study, what we are doing is, by now we have been using 7 controls and 980 cases. We are investigating the temporal cortex. As a typical pipeline of array tomography, we use not 1 but 2 adjacent blocks. We use at least 20 consecutive sections, and we image at least 2 subregions. So to have an idea, we are labeling the physin as presynaptic marker, PSD95 postsynaptic, beta-amyloid, and TMM97. With this, we are doing an analysis typical of array tomography, and then we are looking to this potential interaction with threat that I will explain later. And in this study by now, we are analyzing about 3 million single synapse terminals. So those are the methods. So then the first step, the first results, are to see if we are able to identify this TMM97. Here you can see in this panel the example, a max intensity projection of a control case and an AD case that you can clearly see the flag, and you can see that TMM97 is quite common and widespread. And here what we can do is quantify these 3-dimensional objects that we have in each stain. So I have to say that the results from here on will be shown as a fold change, comparing Alzheimer to control. This is because we are still trying to be 100% sure of the segmentation. So the relation between control and AD, we still we are confident, but we don't want to give like absolute numbers after checking more times the segmentations. So as expected, we can see an increase in beta-amyloid. We can see a slight increase in the density of TMM objects. In blue, you see the Alzheimer cases, but it does not reach statistical significance. And as expected, what we saw was that there is no difference in the total density of synaptic markers. This is already known because this reduction is seen in the vicinity of the plaque. Here you can see a little bit of hole that is giving the beta-amyloid plaque. So the next step is, okay, we can detect TMM97. Let's see if we can find it in the synapses, and even more, if we can find it together with beta-amyloid. So, for example, here we have an example that, yes, at least in some synapses, we can see this. This is a magnification of this max intensity projection of the raw image. And in the columns, what you see are 70 nanometers consecutive segments. It's an example of 6 of them, where we can see TMM97, beta-amyloid, the postsynaptic marker, and the presynaptic marker. And an example of a 3D reconstruction of one of those synapses, where we will have, again, the pre, the post, TMM97, and beta-amyloid in the same synapse. So it looks interesting to go further. And the next step would be to quantify this colocalization. So if we compare again with control, as expected, we will have, this is percent of postsynaptic terminals of PSD95, so postsynaptic terminals with beta-amyloid. There is a slight increase, although it doesn't reach statistical significance. And what is interesting is that we see again this increase in TMM97 localization in postsynaptic terminals in AD when compared to control. Also, even more when we compare those synapses that have both beta-amyloid and TMM97. And this is postsynaptic terminal. What happens if we look to the presynaptic? So something similar. There is an increase in beta-amyloid. Here it does not reach statistical significance for the presence of TMM, but there is a slight increase, there is attendance, and the same for both beta-amyloid and TMM97 together. So from this point, it looks interesting to study the interaction. Also, we wanted to confirm the results with synaptoneurosomes of the same cases. We did Western blots, and by now there are still less cases, but there is also this tendency of a higher amount of TMM97. So taken together, what we see here is that we can see an increase in AD cases when compared to control of the presence of TMM97, and we can see this by synaptoneurosomes, by proteomics, as we saw in the other article, and also by array tomography. And we found it both in pre and postsynaptic terminals. So the next step is to see how close are those proteins, beta amyloid and TMM97. So that's why we are using FRED. So to explain a little bit the Foster resonance energy transfer, let's imagine the synapse. In this diagram, you can see PSD95, beta amyloid, and TMM97. So if those, here we are going to call the donor the fluorophore that is labeling beta amyloid, and acceptor the one that is labeling TMM97. So if they are not close, when we excite the donor and we record the emission of the donor and the acceptor, we will see something like that. We will see a signal in the donor, but not in the acceptor channel. But if they are close enough, it means around 10 nanometers. When we excite with the donor wavelength and we record both the donor and the acceptor emissions, we will see a transfer of energy from the donor to the acceptor that will end up being pictured. So moving to the control, where we can see this, are really important, the controls for this type of studies. In this case, we are doing a sandwich approach. Let's say we are labeling synaptophysin both with the donor and the acceptor fluorophores. So this would be a normal image acquisition. We are exciting with the donor and recording the donor. This would be a normal acceptor image. We are exciting the acceptor and recording the acceptor. But what we are interested here is in this image, that is the excitation of the donor and recording the emission of the acceptor. So here you can see that these 4 or 5 synapses that we see in this single 70 nanometer thick section, we can see it here. So there is spread. Of course, this image needs to be corrected for crosstalk and for direct excitation of the acceptor. But what we can do here is, let's take those synapses and quantify how many of them we see a FRET effect. And if we do this with this positive control, we see that it's around 90%, while if we have only the acceptor or the donor marker, it's much lower. So now that we know that we can detect this FRET effect in synapses, how we did it with DAD. So what we did is, from every case, we are classifying the synapses using a reason of interest. For example, this PSD95, this is a single section again, the segmented image. So we will classify these objects depending if they have both TMM97 and beta amyloid, that would be those PSD positive, or those that are alone or that are only with the donor or with the acceptor. So now, if we quantify this, what we have seen, well, here it reached statistical significance, maybe you cannot see that, but what we have seen is that almost half of the objects where we found together TMM97 and beta amyloid, there is spread, while it don't happen with it's much lower with the rest of synapses. So if we are increasing the number of cases, but if this is confirmed, like it is now, that this is a statistical significant, it's lower than 0.001. So it means that probably they are close enough to generate this effect. So taking together, in summary, we have seen that TMM97 is commonly found in both control and AD cases, that TMM97 is present in both pre- and postsynaptic terminals, that in AD we observe TMM97 in a larger proportion of synapses than in controls, and finally that beta amyloid and TMM97 can be found at the same synapse close enough to see this FRET effect. So just finally, special thanks to the donors and their families, and to Prof. Taresh Perjon and all the team that have been collaborating with this project. And thank you all for this. Very nice.
If anyone has questions?
Marty, interesting presentation. I was wondering if you could comment on the demographics of the subjects or the brains, and whether or not, given the higher prevalence of Alzheimer's disease in women, was there a sex difference that emerged?
That's a really good question. Actually, I don't have the data and I don't remember exactly now, but it was quite balanced. But definitely that's something that we could look at. Of course, the number of cases is not maybe too high to compare for that, but yes, really, we should look at that. I cannot say something now. Actually, we are looking also. We have few 8.3 and few 8.4 cases that may also having some effect on that. So yeah, I cannot answer that now, but it's something that we will definitely look at.
Any other questions?
So Marty, I wanted to, this is Susan, I wanted to put you on the spot a little bit. Last year, Tara had presented results about not only TMEM97 expression across different cortical areas and across different genotypes, ApoE genotypes, but she had also shown some data about PGRMC1. So I just wanted to ask about your plans for potentially looking at an interaction of those two proteins in human brain.
Yes, sure. I mean, it's a really good point, and it's the next one. It's the natural one that we should do now because, of course, it has been shown that TMEM97 and PGRMC1 are forming a complex, so we expect to see PrEP with them together also. But not only this, no, also Street Matter and someone else have shown that they didn't found a binding through PGRMC1. So we need to do this triangle of PrEP test and see if we see the PrEP, and it will give us also an idea of which is the distance that we are talking when we see PrEP, because it's not that common to do PrEP in tissue, and of course we need to be cautious about that. But it's the next step. I hope to be able to answer that really soon.
Thanks very much, Marty.
Thank you very much.
Thanks so much, Marty.
Thank you.
Okay, so our next speaker is Dr. Aladdin Riyad, who's going to speak to us about Sigma-2 receptor, TMEM97, PGRMC1, and LDL receptor complex, and how they're responsible for cellular uptake of A-beta 42 and its protein aggregates. We're very excited.
Thank you, Susan. All right, good morning, everyone. So yeah, just like Susan said, I'm going to be talking to you about the Sigma-2 receptor, TMEM97 PGRMC1, LDL receptor complex, and its role in uptake of A-beta 42. So a little background onto why we started this project was the work that I presented last year and we published last year that showed some strong evidence that TMEM97 and PGRMC1 form a trimeric complex with the LDL receptor, and that this trimeric complex being intact is necessary for the uptake of LDL. And if you disrupt this complex by either knocking out the proteins or pharmacologically targeting them, you end up with a reduction in LDL uptake. So looking at diseases that are implicated by lipoproteins, we decided to look into Alzheimer's disease. Alzheimer's disease is a debilitating disease for the individuals and their families, has a high economic burden, and the incidence rate is increasing with our growing population. Looking in the CNS at what's actually occurring, Alzheimer's disease is characterized by the presence of these amyloid plaques that contain A-beta 42, amyloid protein, and ApoE. The source of these proteins is ApoE is synthesized by the astrocytes, and it's secreted. And its role in the CNS is similar to LDL in the periphery, where it transports lipids in the aqueous environment of the brain and allows for cellular uptake into various cells, such as neurons. The source of amyloid beta is secreted from neurons as a result of proteolytic cleavage of amyloid precursor protein. ApoE has been shown to be able to interact with A-beta, facilitating its clearance from the brain by various methods. The one that we chose to focus on was clearance by uptake, by neuronal uptake via the LDL receptor or the LDL receptor family LRP. So to test this hypothesis, the first thing we wanted to do was characterize a recombinant monomers, oligomers, and fibrils to ensure that we had good preparations of them. Panel A here shows a TM images of our A-beta 482 monomers, oligomers, and fibrils taken by Sophie, a postdoc in her lab. As you can see, the monomers, you don't really see any aggregates formed, indicating that we have a clean sample. Our oligomers, you see small round particles with no protofibrils or fibrils present, and our fibrils, you see clear filaments typical of A-beta 42 fibrils. We also characterize them using a dot blot. All three preparations showed positive reactivity with the 6E10 antibody that recognizes all species of A-beta 42. However, only our oligomeric preparation showed positive reactivity with the A11 antibody, which recognizes is an oligomer-specific antibody. So moving forward, what we wanted to do is the first thing we want to do is test the importance of our trimeric complex in the uptake of these samples, whether they're alone or whether they were in a complex with ApoE. To test this, we used our HeLa cell model system. We had 4 cell lines. We had our scramble Cas9 cell line, which served as our control cell line. We had our PGRMC1 knockout cell line, our TMEM knockout cell line, and a double knockout cell line that was lacking both PGRMC1 and TMEM97. These cells are treated with either monomers, oligomers, or fibrils alone, or these A-beta 42 monomers, oligomers, fibrils in a complex with ApoE3 for 24 hours, and uptake was quantified via ELISA. The first thing that we noticed was that monomers relied on ApoE to get taken up efficiently into the cells. You see very little monomers being taken up, but when they're in the complex with ApoE, you see an increase in uptake. We also noticed that all 3 knockout cells had significantly less uptake than our control cells for monomers in complex with ApoE. The next thing we noticed was that oligomers were able to get into the cells on their own and didn't necessarily require being in the complex with ApoE3 to get into the cells. However, being in the complex, they were still able to get into the cells. We see that the knockout cells had significantly less uptake of oligomers alone or oligomers in complex with ApoE. And finally, we see very little cell-associated fibrils that I'll get into in the next couple of slides here. But we also saw a similar trend where there was less cell-associated fibrils in our knockout cells compared to the controls. So the next thing we want to do is also quantify the amount of ApoE that was getting into the cells, and we did this via ELISA as well. And as you can see, the knockout cells had significantly less ApoE3 alone get into the cells, and they also had significantly less ApoE get into the cells when it was in a complex with monomers, oligomers, or fibrils. Taken together, this indicated that our trimeric complex was important for the uptake of A-beta 42 and ApoE. Next, we wanted to see if we could pharmacologically disrupt the complex and if that played a role in, if we were able to inhibit the uptake of ApoE, A-beta 42 and ApoE by pharmacologically disrupting the complex. We used RHM4 and SW43 ligands for TMEM97, and we used AG205, which is a ligand against PGRMC1. And we quantified the uptake of A-beta 42 and ApoE. And we found that in cells that were treated with these ligands, we see significantly less uptake of all monomers, oligomers alone and in the complex with ApoE3. We saw a slight reduction in fibrils, but not to the same extent, which can be explained in our next slide here. Again, we also saw that when you pharmacologically disrupt the complex, you get a significantly decreased ApoE uptake in these cells. So to tease out what was going on with the fibrils, we used a fluorescent A-beta-42 that we prepared oligomers and fibrils from, and we looked at the uptake over time in our knockout cells. We find that, so panel A is our oligomer preparation, and we find that over time you can see an increase in A-beta-42 uptake, and this increase was, and that the uptake was significantly less in our knockout cells compared with our control cell line. As you can see with the fibrils, you don't really see so much of an uptake, but rather you see it associated with the cell surface. And this can explain some of the results that we see with regards to the uptake of these fibrils. And this is consistent with literature that shows that oligomers and monomers are able to be taken up, whereas fibrils accumulate on the cell surface rather than are internalized. So, as Alzheimer's disease is obviously a neuronal disease, we wanted to look at whether this hypothesis holds up in the context of a primary neuron cell culture system. So to do this, we used day 21 in vitro primary rat cortical neurons. And these neurons, as shown in panel A, are positive for MAP2, indicating that they're mature neurons, and they have a morphology typical of matured healthy neurons. The first thing we wanted to do was to establish whether this trimeric complex exists in these primary neurons. So to do this, I performed a proximity ligation assay, a pairwise proximity ligation assay, where I looked at whether there's an interaction between LDL receptor and PGRMC1. And as you can see, there is. So in using this assay, if you see any fluorescent signal, that indicates that the two proteins are within 40 angstrom of each other, indicating that they're in a complex. When looking at the LDL receptor and TMEM97, you also see a signal. And when looking at TMEM97 and PGRMC1, you also see a signal. So taken together, this data indicates that this trimeric complex is intact in our neuronal cell culture system. So what we did next is we took these neurons and we treated them with A-beta-42 monomers, oligomers, and fibrils alone or in a complex with ApoE2, 3, and 4. Panel A, here's our monomeric data. When we took cells, we also treated them with the RHM4, SW43, and AG205 to disrupt our trimeric complex. When looking at the monomeric uptake, you can see that if you disrupt the complex with either TMEM97 ligands or PGRMC1 ligands, you see a significant reduction in the uptake of monomers alone or monomers in a complex with ApoE2, 3, and 4. Looking at the oligomers, you can also see a reduction in uptake of oligomers alone or oligomers in the complex with ApoE2, 3, and 4. And not surprisingly, we see that there's not really much uptake of fibrils alone or in the complex with ApoE, and that there's no significant difference between our untreated and our treated groups. We also quantified the amount of ApoE that was taken up under these conditions, and we found that when you disrupt the complex pharmacologically, you see an inhibition in uptake of ApoE2, 3, and 4 in the complex with the monomers and the oligomers, but not so much the fibrils, which our fluorescent data helped explain why we didn't really see any difference between the fibrils. So taken together, this indicates that our intact complex is important for the uptake of A-beta-42 alone, which was interesting that it was almost functioning like a lipoprotein in its regards to being taken up via this pathway. Also, we were able to disrupt the uptake of A-beta when it's in a complex with ApoE. So our pharmacological targeting was able to inhibit the uptake of A-beta via two different mechanisms. The A-beta uptake alone or with the complex of ApoE. To show that the actual A-beta 42 oligomers were being taken up by MAP2 positive neurons, we used again the fluorescent A-beta 42 preparation. And as you can see, our untreated group, which is at the top here, had significantly more uptake than our ligand-treated groups, RHM4, SW4, or AG205 treated groups. And that the signal for A-beta 42 did co-localize with MAP2, indicating that the neurons were the ones that were taking it up. So next we wanted to see if the trimeric complex was intact in human brain. We looked at an adult normal human brain from a patient that did not display any cognitive, didn't have any cognitive disease state, didn't have any dementia, and didn't have any amyloid plaques. And our pairwise proximity ligation, our pairwise PLA assay showed that LDL and PGRMC1 were indeed in a complex. LDL receptor and TMEM97 were also in a complex, and TMEM97 and PGRMC1 were in a complex, indicating that this trimeric complex was present in adult human brain. We also looked at a patient that had Alzheimer's disease, and we see that the trimeric complex is also intact in that patient as well. So taken together, our data can indicate that ApoE and A-beta are taken up into the cells via our trimeric complex, and targeting this trimeric complex was pharmacologically with compounds that either target TMEM97 or PGRMC1 can inhibit the uptake of A-beta 42 monomers or oligomers, and can reduce the burden on these neurons, and can potentially be a mechanism by which we can reduce neurotoxicity. And I'd like to thank my lab, and I'd like to thank Virginia Lee and John Trzynowski, collaborators, for providing the brain samples, and thank you.
Thank you, Aladdin. Great job. So I'm wondering if you knock out the TMEM97, right? But have you think about just the mutant TMEM97? Because it's already reported, right? 29 and 56 amino acids.
Right.
If you mutant, you still maintain complex. If those functions still exist.
That'd be interesting to look at. It depends on how the mutation affects the complex. So targeting the TMEM97 pharmacologically, what we hypothesize is that if you disrupt this complex, you're disrupting the uptake. So it depends on what the mutant, if the mutant is disrupted in its the region that binds to the other two proteins, or if it's a different region. That would be very interesting to test.
Okay, yeah, because I'm concerned about the DTG binding. So if you lose the DTG binding, it may still maintain AC205 and the RHM1 function. That will be something worth investigating, right, Bob?
We're not concerned about DTG binding because we don't do DTG binding.
Yeah.
So do you think the A-beta is activating uptake, or does it just go in as part of a sort of basal, constantly operating uptake system? And then do you know what happens to the TMEM97 after it gets taken into the cell?
So, yeah, that's interesting. We haven't looked at the fate of TMEM97 under these experiments, but last year when we were looking at LDL uptake, we saw TMEM97, when it's internalized, we saw that it ends up in the nucleus. So the fate of it is something that would be, we still need to be interested in teasing out. Now, with regards to your first question, A-beta 42 is, what we found is whether alone or in the complex of ApoE, it's one of the mechanisms by which it gets into the cells is the LDL receptor. Now, whether it stimulates further uptake, I'm not aware that it does that. I just believe that this is one of the mechanisms by clearance from the brain is either it can be cleared out of the interstitial fluid to the CSF, or it can be cleared by uptake into the neurons where it's degraded in the lysosome. And literature suggests that that is important to look at because as more of this peptide is taken into the neurons, you can overwhelm the lysosome, increasing the pH, causing aggregation and the disruption of its capacity to degrade amyloid beta, and that can be leading to aggregation and the formation of these plaques and result in neurotoxicity. So.
Could you go back to your previous two slides? This was an N of 1, right, with the PLA?
Yeah, we had just one normal human brain sample and one Alzheimer's disease human brain sample.
Okay, on the bottom row, do you know which cell type you're looking at there?
So we're looking at the frontal cortex for both of the patients.
But is it a microglia? Is it a neuron?
Oh, so that's something
astrocyte?
that we're going to be looking at further in the future.
Well, one thing that stood out to me looking at it, if you go to the previous slide with the normal brain, the co-localization for LDLR and TMM97 seems to be weighted on one side of the cell, so that could be organelle specific. But if you look at the next slide, this pattern seems to switch to LDLR and PGRMC1, but TMM97 is spread all over the cell. I just don't know if you've had any thoughts about what organelle that might be, if it's maybe in the mitochondria or...
So you typically do see that polarized pattern of expression of the LDL receptor on cells. It's an interesting point that you make here that the two do look differently. We are going to be working on teasing out what type of cells they are, and that might also help us understand why we see the pattern that we're seeing. It's just very interesting to see that differences between normal and adult and Alzheimer's disease brains and their expression. Just looking at the expression alone between the two, we see that there's plenty of both proteins. Just like the previous presentation, we saw that in the disease state, you do tend to see a little bit more TMEM97 present than the normal brain. So.
Thanks, Aladdin. I have two questions, and maybe if I won't be greedy, I'll just ask one of them first, and we can talk about the other one later. You mentioned the disruption of the tripartite complex. Did you actually see loss of PLA signal with treatment with the Sigma-2 compounds?
We haven't looked at that. That was our hypothesis that what's occurring. Because whether you target one or the other, you have the same effect of reduction of uptake. But that might be something interesting to see if just treating the cells alone causes a reduction of signal.
Right. And I don't know if there are any other questions. If not, okay. Yeah. I'll ask. The other one, I just wanted to find out a little bit more about the ELISA that you did for the uptake, but we can talk about that later, perhaps.
Okay.
Nice presentation and interesting data. One of the things that I wanted to ask about applies to experimental models that we use. And I was wondering if you've tried to use different A-beta fragments, all of which tend to be cytotoxic, 25 to 35, for example, 31 to 35. They've all been demonstrated to elicit behavioral effects like, cellular behavioral effects like long-term depression, as well as cytotoxic effects. Would you expect to see, or have you determined whether or not these different fragments elicit the same kind of internalization dynamics?
That's an interesting question. We haven't looked at it. The reason why I chose A-beta 42 or let's say A-beta 40 is that it's more cytotoxic to the neurons than A-beta 40. But the smaller fragments, you bring up an interesting point. If they do get into the cells via this LDL receptor complex, I do presume that that they would be decreased in the treated states versus untreated. But again, we haven't tried that out. I'm not sure if they do get into the cells, if this is a mechanism by which they can get into the cells.
If I may just suggest, it might be worthwhile to think about it as a position paper. form the best experimental model that we all use, so we may address the mechanism that's most relevant to the actual disease.
It's a very good point. Yeah, that might be very interesting to look at.
Interesting presentation. Just a quick question, something that I might have missed. I was wondering, for these complex studies, is ApoE isolated or is it part of a lipoprotein when it's being...
So for these experiments, we used ApoE, recombinant ApoE that I lipidated so that it's in the lipidated state, because the lipidation state of ApoE plays a big role on its capacity to be internalized or in its capacity to bind A beta. So yeah, we purchased human recombinant ApoE 2, 3, 4 that I lipidated.
Great, thank you.
Any other questions? This will be the last one.
Okay, so Aladdin, I'm looking at you and I'm looking over at Bob and don't want to preempt Bob's closing comments here, but I was just wondering if you could speculate a little bit on the phenomenon of displacement of oligomers and an increase in the off rate that we observed with some of the Sigma-2 ligands with this. Is that okay, Bob, to ask Aladdin that?
Yeah, go right ahead.
Yeah, so yeah, I definitely am familiar with your displacement. I feel like that's actually a very exciting finding that you guys made. With regards to this, I'm interested to see how that would work. So with our compounds that we tested here, we find that just the fact that you're targeting the protein may cause that complex to fall apart. Now, with regards to displacement, that would be interesting to test experimentally to see what occurs, but I can presume that it would cause some displacement, as you guys have already showed, but yeah, that would be something interesting to look at.
Okay, thank you so much, Aladdin. Our next talk, right before our lunch break, is from Jinbin Xu, who will talk to us about quantitative autoradiography of Sigma-2 receptors in postmortem brain.
Okay. Thank you very much, Susan. My turn. Yeah, it's lunchtime. I feel a little bit hungry. Hopefully, everybody can tolerate and wait until lunch. Okay. So I'm talking about the sigma receptors in postmortem brains, because there are no Sigma-2 tracer for in vivo imaging, so we can only work with postmortem brain to derive the Sigma-1, Sigma-2 receptor density. So it's very encouraging when this paper published 2017 PNAS and identified the binding sites defined by DTG, so that's a mutation. Okay, so Sigma receptors has a Sigma-1 receptor has been well described previously, but the Sigma-2 is less described. So DTG is a traditional radioligand. Yeah, even Bob is not concerned about the DTG, but the DTG is the ligand pharmacologically defined the Sigma-1 receptor and Sigma-2 receptors. Yeah, ITM1, that's a great ligand. Yeah, I like very much, and Bob likes as well, right? So this yeah, ITM1 means ITMAC. So, but there are many questions about this, because this is only validated using DTG binding. And again, so when Alan did his experiment, he also identified PGM-C1 from mass pack. But when they use DTG, there are no binding. Okay. So, so what's the problem with DTG? So those are previous started using DTG binding. That's all from cancer. So Wayne group worked with different cancer cell lines, found sigma-2 density are very overexpressed, and Bob further developed the sigma-2 radioligand as a proliferation marker. It's very useful for cancer imaging and cancer diagnosis. So what has been done for sigma-2 receptor in human brain? So previously, people, that's from 1993, brain research paper, using DTG binding without using sigma-1 ligand to mask sigma-1 receptor binding sites. What was found is actually those imaging from film autoradiography, so image quality not that bad, but the density was lower. So that's from 7 AD brains and 7 control brains. It's interesting to see binding decreased in AD patients compared to controls. Also, cell loss, neuron loss is proportional to DTG binding. So theoretically, we should say those are sigma-1 plus sigma-2 receptors. So the question is, which one last? Sigma-1 last or sigma-2 last? So here they use the 6 nanomolar DTG. So density actually is quite lower, because if you look at the density of the femtomolar per milligram protein, if it converts to femtomolar per tissue, it will be 2 point something, 3 point something. It's about 20 times lower than what we found using our iTRM1 binding. Or it's about 5-6 times lower than thyramicin. Thyramicin is another sigma-2 radioligand. So here is the thyramicin binding. That's not from autoradiography, that's a homogeneous, brain homogeneous binding. So that's a nice curve, even non-specific binding is high, maybe because DTG didn't block very well. That's why it may underestimate sigma-2 receptor homogeneous. But those data, at least, that's close to what we found with iTRM1 binding. I'm going to show you in later slides. So it's curious, DTG binding affinity in brain, it's so low, it's half micromolar. So it's about 20 times lower than reported data from liver homogeneous binding. So what's wrong? Well, based on Alon's paper, PNS paper, maybe there are mutations, or maybe DTG is just not good for measuring sigma-2 receptor in human brain. So that's why we started using iTRM1. Why? Because iTRM1 has been found to be very selective. It's for sigma-2 receptor binding affinity under 5 nanomolar. And for sigma-1, it's 2 micromolar, 2,000 nanomolar. And so that's a binding assay using liver homogeneous. We got 0.66 nanomolar. So also, non-specific binding is very low. So this is an outstanding sigma-2 ligand. So DTG is a very traditional, but iTRM1 is a very novel and very modern ligand to use for starting sigma-2 receptors in cancer, in brain, in all different systems. So yeah, this again, so those are from liver homogeneous. We use the sigma-2 receptor competitive assay and test a series of compounds. Again, we also use DTG under conditions of blocking sigma-1 receptor. So we got a very good agreement. If we put them together, there are some outliers, but most compounds sit in the unity line. That means the pharmacology method using iTRM1 alone is equivalent to DTG pharmacology plus sigma-1 ligand blocking, sigma-1 ligand using the potassium. Okay, so yeah, that's why I say item-1 is good, because much bad high binding affinity. Also, we found it slowly dissociates from ligand. So it's very good for autoradical, because when you do autoradical, you have to rinse. And selectivity is high. So I said that for certain because it's based on KD over KI. We don't really know the KD for item-1 for sigma-1 receptor because binding is too low to be measured. Okay, so we have a very nice beta image system. Actually, currently we have two. So this is very nice because this is digitalized, and it's very fast for treating. Because if you use traditional film autoradiography, on the bottom, that's a film. For 1 month, 3 months, it's the same, not the same slide, that's adjacent section we did simultaneously. We can tell over 24 hours that this image will yield such a nice quality image compared to 3 months film autoradiography. So also it's very quantitative if you measure the digitalized signal over their activity on the standards. So we have this novel tool, and so this is just an example of what we have done previously. Currently, we have over 100 human brains from AD, PD, and with different pathology, with different tau and A beta pathology, so we're working with. So here is the example. It's a very busy slide. So we previously, when Bob was at WashU, we worked together for dopamine D1, D2, D3 receptor, and the dopamine transport, and the vascular monoamine transport too. So there, across all different brain sections, we systematically study those receptor regulation in normal brain and Parkinson's disease or Alzheimer's disease brains. So we have brains available. We also have good radioligand for Sigma-2. Why don't we start the Sigma-2 receptor? Again, so Sigma-1 receptor has very good radioligand potassium. So we can test, we can measure Sigma-1, Sigma-2 receptor. So start from aged human brain, control brains. So those are demographic details for those subjects. We have more females, less males. So, what we have done is process all brains into 20 micron sacks and using radioligand to incubate. We tried the DTG, but we couldn't get a good signal with DTG. But Sigma-1 ligand, the potassium have very nice binding curve. If you increase density, it will saturate, and we can do scan chart. It follows one-side binding model, and we can get the KD value and the Bmax value from the cortex. So we can have a KD. Once we have KD, then we can measure density, use a single concentration based on binding isomer. So this is Sigma-2. This is very, very encouraging. And if you increase concentration, it starts to reach the saturation, and we can tell binding affinity is kind of lower compared to liver homogeneous, because here we got 7 nanomolar, but in homogeneous, liver homogeneous, what we got 0.66 nanomolar. So there are some differences, but still has very good binding, and non-specific were defined using stramacine, 1 micromolar stramacine. So if we do all different regions for Sigma-1 receptor, that's from frontal cortex, basal ganglion, and substantial nigral, we got a very good signal. And we use the haloperidol to define non-specific binding, so we can quantify Sigma-1 density. So those are summarized the data of sigma-1 density is Bmax that's calculated using KD and the apparent binding density. So that's from 11 subjects and the data mean plus SEM. Sigma-2 receptor density is very high, signal is very high. It's displaceable by ceramicin, also other ligands I didn't show here. So we can calculate the total minus the non-spec, that's the special binding, that's the sigma-2 receptor density again, so summarized like this. If we put the sigma-2 receptor together, so we can find the sigma-2 receptor density actually higher than sigma-1 receptor density. It's about 2 times higher other than red nuclear. So most region has a high binding density of sigma-2 receptors than sigma-1 receptor density. So if we plot sigma-1 and the sigma-2 receptor density in the frontal cortex, this is a very, very nice linear correlation. And if we put all the 10 regions together from 11 subjects, we still can say very, very good correlation with some exceptions. Maybe those are outliers, those cases we need to be careful and we need to find out why they're different from those. But such a good correlation between sigma-1 and the sigma-2 receptors. Okay, so yeah, we have other projects. We started tau and A-beta using tracer. So here, this tracer called MK6240. So we can use the same tracer to study those same brains. So it shows promising. It correlates very nicely with pathology. Also has very low non-spare binding. So compared to another radioligand tritium labeled AV1451, it has a lot of non-spare binding. So here's what we try. So again, so those are PET tracers labeled with F18. So once we label with tritium, we can do quantitative autoradiographs using our brain, the brain bank, brains from our brain bank. So the AD, there are 4 subjects. If you only look at AD patients, AV1451, it's very similar to MK6240, but the density is high. So why? Because even use the controls, we still can say AV1451 has a binding. So, but MK has very low binding in control brains, because the control brain still has some tau pathology. So we think from those studies, we choose MK6040 for quantitative autoradiograph. So those are 80 brains we started with our sigma-2 ligand. Now we're using MK to measure tau density. So that's a total binding, that's non-specific binding. So after quantify the density, then we try to plot sigma-1 and the sigma-2 versus tau. At least in frontal cortex, we didn't observe any obvious correlation between both sigma-1, sigma-2 density and the tau. Okay, so let me just try summarize quickly. So again, so ITRM1 is a novel radioligand. It's much better than DTG for binding. Even DTG is a traditional radioligand. So that's why we're still using DTG to compare with ITRM1 for certain cases. And what we found is sigma-2 receptor is extensively distributed in brains. It's sigma-2 density is even higher than sigma-1 density in most brain regions we studied, other than red nuclears. And there are very good co-localization of sigma-1, sigma-2 receptor in brain region. I'm seeing co-localization just at brain level, not in neuron level. And so there are no obviously correlation between sigma receptor and tau in AD frontal cortex. So maybe in the future we will study different brain regions and we will compare beta amyloid, that's a new direction. And So, again, so H1 binding is still high, DTG binding is lower, so maybe mutants, that will be something interesting. Hopefully somebody will be doing this, and we will have more data to share in the future. And the big question is, what are what are the functions of those receptors in normal aging and ADPD? I would like to thank ADRC, Alzheimer's Disease Research Center. They provide us a lot of very valuable human brains, and we thank participants to donate their brains after their disease to help us to advance the research. Also, thank the funding source. Thank you. Any questions?
Thanks a lot. So I'm curious if you've looked at whether the protein levels are correlated with the mRNA levels for TMEM97 or any other of the associated genes across brain regions.
Yeah, that's a good question. So for those proteins, we're working on that, but currently I do not have data to share.
Other questions?
Thanks, Eileen. Great talk, Jim, and really appreciate the work. So in terms of testing interactions with AG205, not a great tool for work with living cells, but, certainly one of the only things we have. Any plans to do that? Do you think it would work? Would it be something that would be worthwhile doing?
So that's a good question, because for Sigma-2 autoradiography, it can test a lot of different compounds, but currently our funding is funded just by like a tau imaging program, a beta amyloid imaging program, neuroinflammation imaging program, but not a Sigma-2 receptor. But I'm considering continuing working on it, because this is the most important project, but the least funded project for me.
So I'm going to tell you a story about the derivation of some of our work that's led to this, and then show you how we ended up asking ourselves the question of what, we've learned a lot about what PGRMC1 does. And I guess we were curious to learn, well, what regulates PGRMC1? One of the things, to cut to the chase, is that we've been spending some time interrogating certain microRNA and learned that LET7I may play an important role in regulating PGRMC1. And by inference, LET7I might influence the protective or death-promoting effects that we're talking about that seem to be mediated by PGRMC1, whether in isolation or in complex with a lot of different players that we're talking about. So just briefly, a couple of disclosures here, to say nothing of the fact that I'll be sharing with you about the potential use of estrogens and progestins in an off-label manner for certain diseases. So I wanted to, by way of introduction, share with you, I guess I'm supposed to stay here, that the average age of the menopause in the United States is about 51. And the mean lifespan for women in the United States, give or take, is about 81. And if you accept the premise that these hormones have very important effects on the brain, to say nothing of the other organ systems that it influences, the question that we asked is, what is the consequence of living a third of one's life in a hormone-deprived state? And whereas most of the history along the lines of this question is focused on estrogen and how estrogen may be neuroprotective or cytoprotective, we recognize that the menopause or the removal of the ovaries in both an experimental model or in oophorectomy represents a precipitous decline in not just estrogen levels, but estrogen and progesterone levels as well. I would like to just make a point that in the human menopause, whereas estrogen and progesterone levels drop quite significantly, androgen levels do not. So androgen levels kind of persist, and I may have time to talk a little bit about the significance of that as the talk concludes. But one of the things that we also started to appreciate, just as much as there's about 2 to 3-fold higher prevalence of Alzheimer's disease in women among men, there seems to be this shift also as a function of maybe a function or coincident with the menopausal transition, where the risk for stroke changes. And this is some work, or actually a review, taken from a review published by Phyllis Wise and Dina Dubal, showing that whereas in this earlier age demographic, you see that men have a higher prevalence of stroke than women, but after the menopause, that shifts such that they've both increased, but there's less of a difference. And whereas the prevalence or incidence issue is somewhat debatable, what is very clear from the clinical literature is women have worse outcomes in response to stroke than men. So the question is why? And so in our thinking along the lines of what's the consequence of living a third of one's life in a hormone-deprived state, and whether or not that change or that reduction in those hormone levels could have made the brain vulnerable, we asked the question, is it possible that postmenopausal estrogen and or progestin therapy may be beneficial against certain age-associated dysfunctions or increases in the risks for certain diseases, to include stroke and Alzheimer's disease? So after making this sort of statement or hypothesis, we went down this path of asking, can estrogen be protective? What's the mechanisms by which it can be protective? And we found lots of literature and our own work to support the notion over the span of about a couple of decades. And this is one piece of work that we did a long time ago, where this was rats that had been overreactomized for 7 months. And what you see is the expression of BDNF. The warmer colors, the yellows and the reds, represent a higher level. The blues represent the cooler colors represent lower levels. And what you can see, there's nice abundance of BDNF in the cerebral cortex and in the hippocampus. But 7 months post-ovariectomy, there was a pretty profound reduction in BDNF levels. Okay, this is in situ hybridization, so we're looking at mRNA. And by the way, 7 months represents about a quarter of a rodent's life. So I want to kind of invoke again that what does it mean to spend a third of a woman's life in a hormone-deprived state? And what happened was, at least with estrogen replacement, and this was replaced 3 weeks after ovariectomy, so it wasn't an immediate replacement, we saw at least partial restoration of BDNF, at least in the hippocampus, but not the cerebral cortex. So one of the things that we inferred from this, or suggested, was that, well, maybe the incomplete restoration was a product of not having progesterone around. So we started to ask, well, could progesterone in and of itself have protective effects? And the short answer is yes. And this is a number of, these bullet points represent work from our lab and from many others that basically talked about how progesterone elicits effect, to say nothing of the fact that progesterone does in fact protect cortical neurons, and in our hands, in an ERK and PI3 kinase AKT dependent manner. So that was great. Progesterone is protective. But then came the Women's Health Initiative. And we were struck by that this very large clinical study seemed to yield results that were inconsistent with all this preclinical and some clinical data that had accumulated. And so we were curious why. And in fact, we came to the conclusion that there were at least 4 reasons that could explain the discrepancy. One was age. where the women enrolled in the memory study component of the WHI, they were 65 years and older. And I submit to you that it's not usual practice to initiate hormone therapy in a 65-year-old woman. And so that led us to ask the question, is there a finite window of opportunity? And I wanted to bring that up again because as we think about PGRMC1 or Sigma-2 targeted therapeutics, that maybe we should put our heads together to ask the question about, is there a window of opportunity by which these Sigma ligands or PGRMC1 ligands have protective effects? And the corollary, however, was that it may not just be biological aging, it could be the duration of ovarian hormone loss that could also be driving the change in responsiveness. But what underlines or what underlies that? And so we started to ask which receptors are involved, and do those receptors change over the course of age? And so, with respect to progesterone, because we've been fairly hormone-centric in our study of the brain, we found that the classical progesterone receptor, that I abbreviate as PR, is essential for progesterone to elicit an increase in the abundance or cellular content of BDNF. So what you see here is the fold increase in, actually six-fold, percent increase in wild-type mice that have the full complement of progesterone receptors in response to progesterone relative to the response to progesterone in animals that have their progesterone receptor knocked out. And you see that there was really no effect in the PR knockouts, but in the wild type there was, suggesting that at least the expression of BDNF, whether it's a function of stability, whether it's a function of de novo synthesis, that was critically dependent on the classical receptor. But clearly that wasn't the whole story, because we came to appreciate that the signaling effects that we had described previously, the activation of the MAP kinase pathway, the activation of the PI3 kinase pathway, they were not sensitive to the progesterone receptor antagonists, the pharmacological antagonists. So we said, well, is signaling and maybe another aspect of BDNF biology that we've been spending a lot of time on, that is BDNF release, how is that mediated? How is that effect of progesterone mediated? And so we started looking at PGRMC1. This is a progesterone receptor membrane component 1. And we thought it was a membrane-initiated event because we could recapitulate the signaling effects with these crude compounds that are progestins tethered to BSA. This bovine serum albumin conjugated progesterone that's connected by a carboxymethyloxime linker. Okay? And so here what you're just seeing is, it might be easier if I did this. This is just showing that we could knock down PGRMC1 with our siRNA approach. And then looking at BDNF release, so this is the amount of BDNF extruded into the media. Under mock transfected conditions with no progesterone added, this was our baseline. When we added progesterone, we got a nice increase in BDNF release. In the siRNA control, this was a scrambled sequence, nothing happens relative to the control. When we applied progesterone, when we just use a scrambled sequence, we still get that nice abundance of BDNF release in response to progesterone. But when we knocked down PGRMC1, we completely lost the effect of progesterone, suggesting that the two receptors might really complement one another. It's the 1-2 punch. You need, on the one hand, the classical progesterone receptor to facilitate and build up the pool of endogenous BDNF. And then there's a separate mechanism that drives the release. But having both together is what drives it. And we've actually explored this mechanism further, and we've learned, quite surprisingly, that this effect on BDNF release is mediated by ERK5. ERK5 is also called BMK1, or big MAP kinase 1. And what I wanted to really show you is something that may influence our experimental design, because lots of us who might study the MAP kinase pathway might use a compound called UO126 or PD98059 to block the MAP kinase pathway. And we had published work with that as well, and lo and behold, though both those compounds inhibit not just ERK1 and 2, but ERK5 as well. So we had to actually go back and ask the question, well, Is it ERK1/2? Is it ERK5? Which is it? And so what I want to show you here is when we knock down PGRMC1 with the siRNA, not only, one, we lost the effect of progesterone on ERK5 phosphorylation, but it had no impact on ERK1/2 phosphorylation, suggesting that the BDNF release that's elicited by progesterone is a product of ERK5. And to support this idea that you need this 1-2 punch, we looked at these astrocytes that have no classical progesterone receptor, but have PGRMC1. When we tried to protect these cells against iodoacetic acid, which is a combined a metabolic oxidative insult, you see reduction in cell viability in response to the insult alone. Progesterone at both 1 nanomolar and 10 nanomolar had no protective effects. But when we transfected the classical PR into it, now you have both the PR and PGRMC1, we were able to see a nice protection, suggesting that the two do cooperate. So Pixar animation, this is not, but just to give you a sense of what we think the working model is for these two. So progesterone can work through the classical receptor to drive BDNF production. But the release of BDNF requires ERK5 signaling, and that BDNF can then act on synaptogenesis, which is a subject of another paper that we published, and have neuroprotective effects, as has been already described. And so to some extent, this has also informed some of the pharmacology, because what we've also found is that whereas progesterone, the quote-unquote natural progestin, is protective, the one that's used in most hormone therapy regimens, MPA, or medroxyprogesterone acetate, is not. And what we think is going on is that it's actually very good at this, that is MPA, but it's ineffective here, or vice versa, because it doesn't allow for the 1-2 punch. In the short term, it's fine, but in the long term, it may have actually depleted. So what we wanted to ask, though, is, but what regulates PGRMC1? And so we turned our attention to some microRNA, and both PGRMC1 and BDNF, as it turns out, doing in silico analysis, have LET7I binding sites. So we asked the question, what would happen if we overexpress LET7I on progesterone-induced BDNF release? And short answer, when we apply the LET7I mimic relative to the control conditions, and here again you see progesterone eliciting BDNF release, when we applied LET7I, that progesterone effect was abolished. Interestingly, that was a product of LET7I and not LET7F. And I want to throw this out here, that the opposite was true when we looked at a model of Alzheimer's disease. So microRNA aren't universally effective. It depends on disease context. And that's another thing that we're learning. Work from Sung Chol in the lab, some of you have met him, have expanded the work from the astrocytes to also look at a model of neurons. And these are differentiated SHSY5Y cells. And what you find is that LET7I not only reduces PGRMC1 expression in the astrocytes, it also does so in this model of primary neurons. A model of neurons, which we've also extended to neurons, primaries. This is also true at the level of the protein. So this is mRNA and this is protein. So, okay, great. LET7I reduces it. But what happens if we kind of apply a age-associated disease, or age-associated pathology, I should say? So stroke. So this is a middle cerebral artery model of stroke. Middle cerebral artery occlusion model of stroke, I should say. And you see not only a reduction in PGRMC1, but you see an increase in LET7I to suggest that the two are related. And indeed, if we just increase oxidative load by applying hydrogen peroxide, you see a very similar sort of thing. You get an increase in LET7I, both in the astrocytes and in the model of neurons. So suggesting that this relationship exists, it's probably a product of disease, so much so that it might set the stage that prevents protective effects of a variety of ligands. Well, so then we ask the question the other way. What if we inhibited LET7I? Could we enhance the protective effects of certain compounds. And since we were focusing on progesterone, we asked the question in a stroke model. What you see here is a TTC stained is a product of stroke. So this is just the middle cerebral artery occlusion. You see a stroke in the cortical area. This is just a rostral to caudal representation from the same animal. And what you see is the stroke causes a lesion. So where you see red, that's healthy tissue. Where you see white is ischemic tissue. Progesterone really didn't do much. In fact, it almost looked like it made it worse. Okay? But when we applied the LET7 inhibitor or antagomer, we got a very robust protective effect. This was seen at the level of motor function as well. And it basically, what it did was it accelerated the recovery. So what you see here is the stroke, what happens in the stroked animal, which is this first bar, what happens with progesterone alone, and then what happens when you combine the antagomer to LET7I and progesterone. So you greatly accelerate the recovery. And over time, most groups sort of show a recovery. Progesterone is now better at day 7 versus day 3, but still you get this dramatic recovery. And now at 7 days, you've got complete restoration of motor function. And this was what really kind of captures it. And I should point out, this is 1 day after stroke. So what you'll see here is these are animals that have been stroked and treated with just progesterone, 1 day. And what you'll see is this stereotypical kind of circling motion because they have hemiparesis, and so they're kind of going to, they have one side of their body is able to move nicely and the other side is not. So they kind of circle. Again, 1 day after stroke, this is the animals that had been treated with progesterone and the antagomer. I couldn't tell the difference between the wild-type animals and these animals. And this is the subject of a patent that we've just recently submitted. So, but what I'd like to suggest is that cellular context matters. I think one of the things that we're very interested in is that neurons and glia interact. And that the expression of the right receptors are critical for progesterone to elicit its full-blown protective effects, and that what regulates some of those receptors include certain microRNA. I know there's a question. I'm just wrapping up now, just to thank lots of people who help us do the work, to say nothing of the funding agencies that allow us to ask some of these questions, and the people along the way that have made critical contributions to the work. Particularly, I'd like to point out Chang Su and Natalia, who played a key role in driving the work that you've just seen, along with Trin Nguyen. So thank you. I'll be happy to take any questions you have.
Just real quickly, were the mice that were ischematized, were they mixed male and female, or were those only female?
No, those were just female.
Those are only
Yeah, yeah. So that's actually the phase 2, to compare this efficacy in males and females. Yeah.
Thank you. So I'm curious if you've compared isoforms of BDNF. And I guess what's motivating my question is, especially with some of the earlier slides you showed of in vivo BDNF differences, if some of them could be downstream of differences in neural activity.
Yeah, so we've tried to approach that one of two ways. We've looked at pro versus mature. That's one, and we see favoring of mature in response to the progesterone through PGRMC1. If we actually knock down PGRMC1, we lose that effect. The other is to actually look at in a 2-cell model where the hypothesis is progesterone works through astrocytes to release important growth factors to have protective effects on neurons. And that data would suggest that that's an important route. That's not to discount direct effects of progesterone on neurons, but a major effect, at least on the synaptogenesis side of things, appeared to be mediated by BDNF release from glia, then acting on receptors on the neurons to elicit increases in synaptogenic markers.
Okay, so great work. So I wanted to follow up on a comment you made about the I versus F being the opposite in Alzheimer's disease.
Good question. So that's actually the basis of a grant that goes out in February, because I'm not sure I'm smart enough to figure that out without having some funds to do the study, because in the 5X FAD mouse, it was LET7F that was increased, not LET7I. And yet, in our stroke models, it was LET7I that seemed to play the dominant role in terms of just the consequence of pathology. I can't speak to the intervention just yet, but the pathology seems to have a differential influence on the LET7 family. So is it possible that on the one hand, maybe amyloid is involved, or its processing is involved in a different way? Our very provocative idea that I want to just throw out there for critique is that perhaps any intervention that engages the BDNF system might benefit from inhibiting one or more of the LET7 family members. Because if BDNF is indeed so important for a variety of protective effects, whether it's effects on the cytoarchitecture of the cells, whether it's effects on long-term potentiation, if it's effect on intracellular signaling, differentiation, what have you, maybe we could augment some of the therapeutic effects of some new, or maybe even some of the ones that already exist, interventions, by enhancing the stage that involves the BDNF and its players.
Thank you. So one last question, if I could. So you mentioned earlier the androgen story. So maybe you could talk a little bit
Actually,
more.
I have to owe a debt of gratitude to Wayne, because one of the things that we've been unable to understand is, and I'll share with you the perspective. What I don't have time, because it's almost like a separate story, but it's parallel, is that we have identified an androgen receptor mechanism that it resides at the plasma membrane that seems to be sensitive to PGRMC1 inhibition. That is to say, but in this case, in the context of this membrane androgen receptor that we've been calling it, and actually membrane-associated androgen receptor, to distinguish it from Peter Thomas's MAR and other folks, like AR45. In this case, when we activate through a PGRMC1 dependent mechanism, it actually favors cell death. Whereas here, in the neurons and the astrocytes, in a PGRMC1 dependent way, progesterone promotes survival. So based on some of the players that we're talking about, I'm really interested in having some follow-up conversations to help us understand why the difference. Because on the one hand, that death-promoting effect, we can mitigate by using siRNA to PGRMC1 or AG205. And I appreciate that there's some limitations of AG205. But these are also conversations I wanted to have with Bob, because I think some of the compounds you all have and some of the science that you've already built could teach us. So that's where that's coming from.
Thank you.
Other questions?
Thank you very much.
Thanks, Dr. Singh.
Okay, next I'd like to introduce Dr. Carmen Abate, who is going to talk to us about modifying Sigma-2 receptor lead compounds to produce differently purposed Sigma-2 targeted agents. Thank you.
Okay. First of all, I'd like to thank Susan and her crew for organizing this meeting and Loyola University for hosting us. My talk will not have much to do with neuroscience, but of course will have to do with Sigma receptors. I will talk about structural changes that we made on our lead compound PB28, and the structural changes led to highly Sigma-2 receptor selective agents. This is the, sorry. Okay, I will not use laser, I will use just the arrow. Compounds that are endowed with collateral sensitivity that are useful in the treatment of resistant tumors. These changes also led us to adopt a multifunctional approach to eat diverse tumors and to develop imaging Sigma-2 receptors probes. We started with Sigma receptors in 1995 when, through a multi-receptoral investigation on 5-HT1A receptor ligands, we ended up with this structure that is a tetraline linked to a piperazine that was also endowed with sigma receptor affinity. Therefore, this compound became our lead compound for the development of structural affinity relationship studies that led few years later in the generation of PB28. PB28 is this tetraline connected by a propylene linker to a cyclohexyl piperazine and displays subnanomolar affinity for both sigma receptor subtypes. This was one of the highest sigma receptor ligands for the time. And of course, a number of studies came after that showed how this compound had cytotoxic activity in several tumor cell lines and also in vivo a moderate activity in pancreatic tumor models. This compound was selective for other subtypes for the CNS, but was unselective for one of either the two subtypes. So it had nanomolar affinity, equal nanomolar affinity for both the sigma subtypes. Therefore, it was unselective. Therefore, structural changes were made on PB28, and I will summarize these structural changes that were to the tetraline or to the basic portion, which led to this interesting for the as biological profile compounds, which are F281, PB221, and F264 that came from PB28, and these compounds, together with literature reference compounds such as ceramazine, prompted us to develop a kind of mixing ligands approach, meaning that we mixed the portion of these molecules to obtain a novel class of compounds. So we mixed the hydrophobic portion of the molecules that I showed with the basic moieties, that are the moieties responsible for the sigma receptor affinity, and we obtained compounds with low nanomolar sigma receptor affinity, Sigma-2 receptor affinity in particular, and with cytotoxic activity in breast adenocarcinoma MCF7 cells and the resistant counterpart that overexpress PGP, the pig glycoprotein efflux pump. These compounds were endowed with collateral sensitivity properties. That means that their activity, some of them, had an activity which was higher in resistant cells than in the wild-type counterpart, which is the opposite. That's what usually happens. And these compounds were also a potent modulator of PGP. What is collateral sensitivity? Collateral sensitivity is this property that I said that is characterized by a higher activity, cytotoxic activity of compounds in resistant cells than in the parent counterpart. There are different hypotheses that account for collateral sensitivity. One has to do with the perturbance of the cell membrane, but the other hypotheses all deal with the energy stores of cells, meaning that compounds that are able to deplete the energy stores of cell can be endowed with collateral sensitivity. So can have this activity of hitting, of killing more the resistant cells than the parent cells. The compounds from our mixing ligands approach with the best activity were F397 and F408. Both these compounds were endowed with nanomolar, and in particular F408 subnanomolar affinity for Sigma-2 receptors, and an important selectivity towards Sigma-2 receptors versus the Sigma-1 counterpart. They were both potent PGP modulators. They were able to revert the resistance, the PGP-mediated resistance in co-administration with doxorubicin. So doxorubicin could be active again in co-administration with this compound. And they were both endowed with cytotoxic activity in breast, lung, and colon carcinoma, wild-type and resistant counterparts. In particular, F408 was very active in resistant cells. And of these two compounds, F397 was not endowed with collateral sensitivity, but it was able to kill tumor cells. was not able to kill more resistant cells than the wild-type counterparts. And in fact, F397 was not an ATP depleting agent, did not deplete ATP, because it is a PGP inhibitor. So it inhibits the pump without activating the efflux, and so without activating the ATP-mediated efflux of the pump. Whereas F408 was a collateral sensitivity agent because it was able to deplete ATP, since it is a PGP substrate with micromolar submicromolar activity for PGP. Also, ceramycin was shown to be a PGP substrate, and also ceramycin had these collateral sensitivity properties. These properties were lower compared to F408, but also ceramycin was able to kill more resistant cells than the parent counterpart in breast, lung, and colon carcinoma cells. Therefore, the hypothesis that can account for the collateral sensitivity of these two molecules can be due to the fact that these molecules can activate a futile ATP cycle because they are PGP substrates. We wanted to push this concept further. What we did was to attach a metal chelator portion to the Sigma-2 PGP pharmacophore of these molecules in order to have more potent anti-tumor agents. Sigma-2 moiety was used to direct versus tumors and possibly exert cytotoxicity, whereas PGP was inserted in order to circumvent the efflux or to induce collateral sensitivity. Metal chelation, of course, in particular chelation of iron and copper, is able to hit the redox state of cells, and so to deplete the energy source of cells. In order to obtain these molecules, we used two metal chelator portions, thiosemicarbazons and pyridine-pyranones, that are responsible of the metal chelation. To these moieties, we attached a linker binding at the end a basic moiety. Our golden bullets were cyclohexypiperazine and 6,7-methoxytetrahydroisoquinoline in order to hit PGP and Sigma-2 receptors. The thiosemicarbazons were endowed with high Sigma-2 receptor affinity, PGP modulator properties, and metal chelators, whereas pyridine-pyranones were not able to bind Sigma-2 receptors, although they were metal chelators. Therefore, we focus our attention on thiosemicarbazons. And the most interesting thiosemicarbazons were PS3 and MLP44, which had cytotoxic activity in breast, lung, and pancreas tumor cells. They were able to increase reactive oxygen species and to induce mitochondrial superoxide radical production. These molecules were challenged with the corresponding molecules that do not bear the Sigma-2 targeting portion. So the Sigma-2 targeting portion is this linker with the basic moiety at the end. The ACTO1 molecule did not have this portion targeting Sigma-2 receptors and PGP. These molecules, non-targeting Sigma-2 receptor, was endowed with cytotoxic activity. However, it did not produce increase in ROS, and it did not produce mitochondrial superoxide radical production. In vivo, these molecules were tested in a pancreatic tumor model, and all the three molecules, either targeting or non-targeting Sigma-2 receptors, were able to reduce tumor volume, but the targeting moieties, the Sigma-2 receptor targeting moieties, did not show signs of toxicity, whereas ACTO1, which was devoid of the Sigma-2 receptor targeting portion, led to pulmonary metastasis and nephropathy. The results obtained by these molecules show that the Sigma-2 PGP targeting moieties is not needed for cytotoxic activity, and we show that both in vitro and in vivo. However, Sigma-2 targeting activates diverse pathways, cytotoxic pathways, and also reduces toxicity. Additionally, different basic moieties are able to lead to different pathways, and diverse cancer cells are sensitive in a different way to these different pathways. Therefore, these findings can lead to can be seen in a view of a personalized medicine approach. Another molecule from the thiosemicarbazone, which was endowed with a very interesting activity, was MLP55. This molecule was a PGP inhibitor with a submicromolar activity for PGP, no cytotoxic activity, and it was used to revert doxorubicin resistant in transforming doxorubicin, that is a BBB impermeable drug, into a BBB permeable drug. And we showed in an in vivo model that this molecule was active in a glioblastoma derived from a patient. So this glioblastoma is rich in stem cells, and this molecule, MLP55, was able to make doxorubicin active in this tumor more than the temozolomide. It is the drug that is used as compound for treating these tumors. The compound that I was going back, I talk about this compound, PB221, which is a derivative of PB28. This compound was very cytotoxic in neuroblastoma cells. And also this compound was tried in vivo in both pancreatic tumors and brain tumors. In pancreatic tumors, it had a moderate action, whereas in brain tumors, in vitro, it was able to reduce migration and invasion of glioma and astrocytoma cells. And in vivo, both in an ectopic and orthotopic model, this compound was able to reduce tumor growth and to increase survival. Therefore, PB221 is being proposed as a potential drug for brain tumor treatment. Going back to our mixing ligands approach, we, taking inspiration from Bob Mack's benzamides, we developed a series of benzamide. And from our structural affinity relationship studies, we hypothesized that these benzamide interact with Sigma-2 receptor through an intramolecular hydrogen bond, meaning that maybe they assume this conformation to bind Sigma-2 receptors. In order to test this hypothesis, we have frozen this intramolecular hydrogen bond, creating this isoquinolinone structure that as the benzamide portion frozen in this conformation, basically conformation. And later in the years, we also developed a quinoline and isoquinolinone structure. The compounds obtained through these studies were endowed with low nanomolar Sigma-2 receptor affinity, excellent selectivity, and not cytotoxic action. So these compounds were not cytotoxic. The best compounds from this series were F390 and FS28 and Fa10. They are, as you can see, all endowed with nanomolar affinity for Sigma-2 and micromolar affinity for Sigma-2. Therefore, they are Sigma-2 selective. And they've been used as blocking agents for Sigma-2 receptor biological assays because they are not cytotoxic and they can be safely used to block Sigma-2 receptors when we want to block this action. And they've become also leads for Sigma-2 receptor imaging agents. Therefore, F390 and PB28 have been transformed in order to produce fluorescent small molecules. After structure affinity relationship studies, we understood that it was better to replace this methoxy group with an alkoxy linker, bringing at the end a fluorescent tag. Therefore, we put a fluorescent tag either on PB28 derivative and on F390 derivatives. We obtain high affinity and selective Sigma-2 receptor small fluorescent molecules, which were used in flow cytometry to obtain binding data for either novel Sigma-2 receptor ligands or reference compounds that validated these assays. Therefore, these compounds may replace the use of radioligand binding assays in a safer and cleaner, from the green point of view, assays. We also, so these are fluorescent small molecules hitting Sigma-2 receptors. We applied this same strategy to obtain nanoprobes, so big molecules, in which the small fluorescent tag was replaced by a big luminescent quantum dot that emits in the red. These luminescent quantum dots are endowed with superior optical properties. They have high photostability, long lifetime, wide range of absorption and emission. You can modulate the properties, emission and absorption wavelengths of these molecules. The question was, are these molecules with this big portion able to specifically bind Sigma-2 receptors and to enter cells? The answer is yes. We showed that these molecules were able to enter cells and to specifically bind Sigma-2 receptor. Therefore, these molecules hold promises for the development of Sigma-2 receptor nanoprobes for the imaging from a diagnostic and also therapeutic point of view, because nanoparticles can be charged with drug payloads that can be specifically delivered to Sigma-2 overexpressing cells and deliver the drug payloads. In summary, I have showed that structural modification on Sigma-2 lead compounds, either our lead compounds or reference lead compounds, have led to Sigma-2 receptor imaging agents, small molecules, and nanoprobes, and to promising anti-tumor agents that we have shown also from preclinical models, in particular for pancreatic and brain tumors that are very aggressive tumors. The big question is Sigma-2 receptor binding of these molecules responsible of the activity of these ligands? Of course, we want to answer to these questions, and we are all working for that. However, we have shown that Sigma-2 targeting reduces the toxicity of these molecules, maybe because of less off-site effects, toxic off-site effects. And the Sigma-2 targeting also activates alternative pathways and in the cells where they activate cytotoxic activity, and this can be seen in the perspective of a personalized medicine approach. I thank you all for your attention, and I want to acknowledge my colleagues, in particular Mauro Niso, Maria Alessandra Contino, Maria Laura Patti, and Chiara Riganti, who are all responsible of the biological handling of our studies, and my boss, Professor Francesco Berardi. And thank you all.
Thank you, Carmen. Very nice work. I know you have cell lines that have PGRMC1 knockdown, and I don't know if you have TMEM97 knockdown, but I'm wondering if the cytotoxicity effects that you're looking at still have toxicity with either of those proteins removed.
Okay, so we have shown that removing PGRMC1, at least in the cell in which we removed it, that is MCF7, the activity of the compound stays the same. So they still are cytotoxic. With TMEM97, we are working on that, and I cannot give you a straight answer right now, but next time.
I was wondering, for any of the compounds that have good Sigma-2 affinity, but that don't kill cells, did you see anything in relation to MTT stimulation? Or I don't know if you're using the MTT assay or not.
Can you repeat the last part of the question, please?
So in your compounds that have good Sigma-2 affinity but are not cytotoxic,
yeah.
Did you see any evidence for increasing MTT reduction or anything about that?
No, we didn't check that. However, we do the MTT assay, and there was no activity on that. So we.
Okay, thank you, Carmen. So I wondered if you could expand a little bit on the personalized medicine
approach
comment that you made. So if you could talk about the phenotypes of the tumors in terms of the expression and what you would expect to see with the different ligands.
Yeah. Like in particular with thiosemicarbazone, we have seen this, that thiosemicarbazons always hitting Sigma-2 receptors. When you use these compounds in pancreatic cells, you see that in pancreatic cells, the same compounds in different cells, such as, for example, I don't know if you are familiar with pancreatic cancer cells. However, PANC01 is a very aggressive pancreatic tumor. And Miapaka 2, I don't know how you call it, is another one. So the same compound in the two cells is able to activate different pathways. And the cells are sensitive in a different way to these pathways. Therefore, if you know what are the pathways that you have to hit in order to obtain the cytotoxic effect of the compounds, then you know what kind of tumors you can treat with that compound or not. So in this sense, that is the personalized medicine approach, knowing the phenotype of the tumor and what is the pathways that compound is able to activate in the tumors. And this is particularly visible in these pancreatic tumor cells that we have seen, that we are studying more than the others. So maybe you can have the same effect in the other cell lines, but we are more focused on pancreas and so.
Quick question, Carmen. Do you know what the sex of the patient from which the tumors originated?
I guess a male.
Female?
Male. I'm not sure. I have to check because Chiara Riganti is the last name is the one responsible. I think is a male.
Both male. Okay.
I will tell you later anyway to be sure. So I will.
Any other questions?
So it gives me great pleasure to introduce my colleague Nick Izzo, who is going to talk about metabolomic evidence of disease modification in mild to moderate AD patients by Sigma-2 antagonist CT1812.
I don't know. I'm not sure that.
You can take your badge off because it'll.
Okay, put that here. Is that? And then.
Making sure it's on.
Okay. Does that work? Yep.
Nope.
Does that work?
Yes.
Okay, thank you. Thank you, Susan, and thank you, everybody. Yes, I'm going to talk about metabolomics a bit, but I'm going to take you on a little bit of a journey. I will get to metabolomics. It might not seem that way at some point. So as I'm sure you all know now, Cognition Therapeutics has been engaged in developing small molecule therapeutics for Alzheimer's disease. And while there are a number of different approaches that have been taken that focus on amyloid beta, from monomers to things that target fibrils, we've been focusing on A-beta oligomers. And Susan might elaborate a little bit more on some of these other approaches and their failures in the clinic. But we've been focusing on A-beta oligomers, which are the most toxic species of amyloid beta. What we did is we targeted phenotypic assays to look at cellular effects of A-beta oligomers and screen for compounds that would counteract these cellular effects. And what we found are small molecules that would displace A-beta oligomers from their binding sites on neurons and block downstream effects. So I think you've probably seen this data before. Susan showed some of it earlier, but this shows some of the ways that we've looked at three different complementary methods that we've used to look at displacement of A-beta oligomers. On the left, you could see some in vitro results from cultured neurons. How does this work? Oh. Okay. This is in cultured neurons. And you see immunofluorescence of A-beta oligomers. And then when CT1812, which is our compound that is in the clinic, is put on after the A-beta oligomers, you displace a vast majority of the oligomers. And here you can see a saturation binding curve, just A-beta alone, and then in the presence of CT1812, and you see the decrease in binding at all concentrations of oligomer. We also have looked in tissue from Alzheimer's patients. So these are postmortem brain samples. We looked in tissue surrounding the dense core plaques and look at immunofluorescence of A-beta, and you can see from this tissue when the frozen sections, when they're put in, thawed and put in PBS in vehicle or A beta, you can see a dose-dependent decrease in the amount of A beta bound to that human brain tissue. And then I think a lot of you have seen this data before. This is an in vivo situation where we have, this work was, the second panel was done by Tara Spires-Jones and Chris Henstridge in Edinburgh. The last panel, this panel here is in vitro mice where working with John Cerrito and Carlo Udi at Washington University, they've developed what they call a microimmunoelectrode. This is a 5-micron carbon electrode that's coated with A11 antibody. You've heard a little bit about A11 antibody earlier. The antibody attracts A beta oligomers to the electrode, and then an electric pulse oxidizes the tyrosine in the A beta, and you can measure through like standard electrochemical methods the concentration of A beta oligomers. In this panel in the blue, the electrode's placed in the hippocampus, so it's interrogating the interstitial fluid in the hippocampus. A baseline is measured. The electrodes could measure concentration on a minute-by-minute basis. Drug is administered to the animal at where this vertical line is, and then you could see an increase in the soluble A beta oligomers in the interstitial fluid in drug-treated versus the vehicle-treated animals, where you see this maintenance of a very steady baseline. What's more, if we take this electrode and you stick it in the lateral ventricle and look at what's going on in the CSF after a drug is given, you can see an appearance of oligomers in the CSF. So we've shown 3 different methods here, in vitro, ex vivo, in vivo, of displacement of A beta oligomers, and what's more, clearance out of the brain into the CSF. So we know that there's a lot of literature showing that most of the toxic effects that happen in Alzheimer's disease are downstream of A beta oligomer binding. One of those things is disappearance of synaptic spines. So this just shows more in vitro immunofluorescence here. And here you see, this is a neurite in culture that runs along horizontally here. You see red is A beta oligomers detected by immunofluorescence. The green is a synaptic marker, synaptophysin. So you can see in this case when A beta oligomers are added, you see a lot of oligomer bound and very low number of synapses. When the CT1812 is added, in this case was for 24 hours after the A beta, you see very little A beta binding, and you see a large increase in the number of synapses. And then downstream from that, we've done behavioral studies in transgenic animals and done a number of different behavioral memory tests, so fear conditioning and Morris Water maze, and you can see here in APPs, so these are mice with human APP with a Swedish London mutation. You can see the deficit in both assays in the red bars in the transgenic animals, and that's restored to normal when those animals are given 10 mg per kg of CT1812 once a day. In this case, it was for 9 to 10 weeks. And more importantly, it's important to notice we've also looked at the wild-type animals with the drug, and you can see in the last bar, there's no change in their performance in these behavioral tests, showing that we have a very specific mechanism that is targeting A beta related deficits. It's not a general effect on cognitive ability. So, of course, the reason why we're here, the reason we've brought you all here together is that our compounds are Sigma-2 ligands. This shows some data that we published in 2014, working with Bob Mack and Jinbin, and they showed very nicely here. This is human brain tissue autoradiography with Sigma-2 ligand. You can see these were actually 2 early analogs in our compound series. You can see dose-dependent inhibition of the displacement of the Sigma-2 ligand. And then this is our CT1812 here. In this case, this is in human Jurkat cells, and you can see the displacement of DTG with a binding affinity of about 8.5 nanomolar. So we've repeated in vitro binding in a number of different systems, PC12 cells, rat liver membranes, and human TMEM97 cloned into HEK cells, and we always see this single-digit nanomolar affinity pretty consistently. So the other part of the story here today is PGRMC1, and this again is work that we published several years ago where we knocked down PGRMC1 in our cultured cells with siRNA, and here in panel A you can see the binding of the A-beta oligomers in cells treated with the siRNA. You can see a drastic knockdown in the oligomer binding compared to non-targeting siRNA in panel C. And we actually saw a nice correlation between the amount that we were able to knock down the PGRMC1 and the amount of binding. And interestingly, it turned out that the most we ever could get was about a 30% knockdown of PGRMC1, and that seemed to eliminate 90% of the binding. So there seems to be a very tight coupling between the amount of PGRMC1 there and the binding of oligomers to the cells. So of course, one of the big questions we had are these two proteins that are involved in the Sigma-2 receptor, are they actually where they need to be in order to be mediating this effect on A-beta binding? We knew from our previously published work, this is a neurite along here, and you see labeling of synaptophysin and A-beta oligomers. This is actually a pretty short-term incubation where you wouldn't see downregulation, and you can see some of the yellowish overlap between the A-beta binding with synaptophysin. So we've labeled cells with antibodies for TMEM97 and PGRMC1. So these are, you can see, well here you can see a composite image of MAP2 labeling, and the PGRMC1, or TMEM97 is expressed at high levels in the soma and also out in, along the neurites in lower levels, pretty diffusely throughout the soma, throughout the neurites. PGRMC1 we previously looked at also, and we could see a similar pattern. It's pretty bright in the soma and then out along the neurites. In the system we're looking at, it's really hard to see any kind of punctate labeling here. It's pretty faint labeling. So we've gone to other methods, which you've heard a little bit about today also. So proximity ligation assay, which Aladdin was very nice to give us some tips on. So here you see in our cultured neurons, this is looking at a PLA interaction between TMEM97 and PGRMC1. So what you see in the top are MAP2 labeling. You see the nuclei and MAP2 labeling, and then labeling of synapses. In this case, we use synaptotagmin 1. It just gave us a nice, a little bit crisper labeling. And then the PLA reaction. And you'll notice you can see the PLA labeling that runs along the course of the neurites here. You see a lot of other things here. In these cultures, there's a very fine network of extensions of neurites that don't necessarily show up in the MAP2 staining because they're so faint unless you really overexpose the image. But they're still running in lanes here. And then if you look at an overlap, or you look at, compare where the PLA interaction and the synapses are, you can see a very remarkable overlap in the labeling. So it's about an 80% overlap, although we do see some instances of both PLA interactions that don't have associated synaptic markers and the opposite, where we see some synaptic markers where there's in PLA. And we don't really know if that's an absence or just below our detection limit here. We're not doing a very high resolution type work like you heard earlier by Marty Colomb, but that would be it would be very good to see what they're seeing with their array tomography with co-localization. So this is work that was done by Kelsey Sadlick in our lab, and I just want to advertise her poster on Sunday afternoon, so please stop by and talk to Kelsey and see some more details about this work. So really what we've seen are, the receptors for our drug are located in the location where A-beta oligomers are binding and where we see the drug act to release the oligomers. So what do we think is going on? So this is just a schematic of what we think is going on. So and up here you see a synapse, and you see presynaptic area, and postsynaptically that's where we believe the action is going on with A-beta binding. We haven't really localized these to postsynaptic membranes. We don't have the resolution with what we're doing right now to see that. But if you look at what's in there, there's a A-beta binding site, which some very good work reviewed recently by Steve Strittmatter has identified 3 proteins that are pretty there's very strong evidence for being binding sites. So LRB2, which is also known as PIR-B in rodents, no-go receptor 1, and the cellular prion protein. There are probably other things involved here too. Maybe LDL receptors are part of it. Be very interesting to continue exploring that. Then we have this receptor complex, TMEM97, PGRMC1, which interact with each other. And you notice we've also left a little box here, a little mystery box here, allowing for the possibility that there's yet another protein that might be involved with these 2 proteins that, not that we have any evidence for such a thing, but you never know what the future holds in store for you. So this is the situation normally. These proteins are floating in the membrane. And in Alzheimer's disease, you get an accumulation of A-beta oligomers. They come in and they bind to some receptor complex. And our hypothesis is that this PGRMC1, TMEM97, Sigma-2 receptor is interacting with that binding site and stabilizing that oligomer binding site. I think you've seen a few bits of evidence here today that in Alzheimer's disease, the Sigma-2 proteins accumulate or seem to increase. We had published some data previously suggestive of that. And I think one possibility is, this proteins, they have normal turnovers, but if they're stabilized in a receptor complex with A-beta oligomers, then perhaps they're getting kind of stuck at the membrane and they're not getting turned over like they normally would. That could be part of an accumulation here. But then when you add Sigma-2 ligand like CT1812, the 2 jumped down there, you're altering the conformation of this receptor complex, which is kind of breaking its association with this binding site, and now it's no longer stabilized into a complex, and the oligomer comes off. And then downstream of whatever's going on gets turned off. So, like I said earlier, we know that many of the pathological effects in AD have been shown to be downstream of A-beta oligomer binding, and well, I wrote here, the question arises. That's a rather passive way of putting it, so I'll more assertively say, I am raising the question that how many of these pathological downstream effects are due to sequestering or locking up PGRMC1 and TMEM97 into some complex. So we've heard a lot of evidence about a lot of biochemical pathways that these proteins are involved with. So they have normal functions that they should be doing in a normal state. And in Alzheimer's disease, if they're being locked up and kept away from doing their normal work, is that causing some of the effects? And then the next question is, can these abnormalities be relieved by putting in a drug that restores things back to their normal state? This is a slide that I borrowed from Mike Cahill's talk from last year, and he's going to talk more detail about this, I'm sure. But it just shows a lot of the places where PGRMC1 is involved, particularly in cholesterol and lipid metabolism. So you can look at Mike's paper where he talks about all these mechanisms, but you can see that there's a lot of different work, and Mike's done an enormous amount of work on reviewing this literature and coming up with some very beautiful hypotheses about an overall regulatory system here. Besides PGRMC1, we know that TMEM97 affects lysosomal lipid storage and cholesterol transport. We've seen Aladdin's work on cholesterol uptake. So there are a lot of places where these proteins are involved in lipid chemistry, cholesterol chemistry, and a lot of places where these things can be disturbed in Alzheimer's disease. So it happens that this has been looked at. So this is a paper published a couple of years ago, Toledo et al. And this is Rima Kadura-Dok's group down at Duke, and she was really one of the foundational people in the study of metabolomics. So in this paper, there are some nice things here. So they say, complex disease such as AD arise from alterations in multiple genes, proteins, and metabolites. So there are wide-ranging effects that are being altered by the disease pathway. And we know that a lot of metabolites in general are interrelated with each other in a number of different ways through pathways and cofactors and intermediates. So they did this study of 905 patients from the ADNI-1 cohort. So this cohort, there's a vast amount of data on cognitive changes, imaging changes, and biomarkers. And they did a metabolomic study using a kit from a company called Biocrides that can measure, it's called the P80 kit, but actually measures 188 metabolites. So there's just a little bit about that kit. So it uses an extraction process and then mass spec, double mass spec method for measuring a number of different categories of metabolites. So you can see the interesting ones here. They're acyl carnitines that are involved in energy metabolism and fatty acid transport, amino acids that are involved in a lot of different things, hexoses and carbohydrate metabolism, phosphatidylcholines, lysophosphatidylcholines, and sphingomyelins. The later two are particularly involved with neuronal damage because phosphomembranes are such an integral part of normal neuronal functioning. So all this data is available through the ADNI consortium in addition to the stuff that they published about. So it'd be interesting to look. We thought it would be interesting to look at that type of thing. So we happen to have some samples from clinical trials that we're doing. So I think it was Susan mentioned earlier, we're now in phase 2 studies of CT1812. The first 2 trials that have been completed, one was a phase 1 clinical safety trial, and that was published the beginning of this year. And then our second study was a safety trial in mild to moderate AD patients compared to the first study was in healthy normal patients. So this study was a 28-day blinded study, double-blinded, placebo-controlled study, 19 participants with mild to moderate AD. They were treated once a day with placebo or 3 different doses of drug for 28 days. So there were a number of safety measures that were made along the way and pharmacokinetic measurements, but we also collected biomarkers at screening from plasma and from CSF. So it's screening before the study and then at the end of the study. And I'm going to tell you about some of the measurements we made from the plasma samples, and Susan's going to tell you some of the stuff that was done from the CSF samples. So looking at our patient samples, what we found when we compared all the treated patients together versus placebo-controlled, we saw that there were 11 metabolites that were significantly increased in the drug-treated patients. Now, I should point out that none of these were significant by when you correct for multiple comparisons. This was a pretty small test. It was a pretty short duration. Yet, what we see is this remarkable pattern in that if you compare the results in this study with what was published in Toledo et al., you see that in the drug-treated patients, 10 of the 11 metabolites were actually moving in a direction opposite of the way they move in Alzheimer's disease. So it looks like they're moving a lot of these metabolites in a therapeutic direction compared to how they're changing in Alzheimer's disease. Another way to look at the data is if you look at the metabolites that Toledo saw significantly change, and that's in the red there. They found 18 metabolites overall that were significantly altered in Alzheimer's. You can see some of them are pretty small changes, but they tend to be tightly regulated, so they were statistically significant. And then if we looked at what happened in our study with those same metabolites, we see 12 of those 18 metabolites moving in an opposite direction than they were changing in Alzheimer's disease. Now, if we look at particular classes of metabolites, one set of metabolites that looked pretty interesting were long-chain polyunsaturated fatty acids. So there have been a couple of studies that bring these metabolites to that show them to be of interest. And I should say another thing about our study is that the Toledo paper, all the patients that were looked at were fasted overnight before samples were taken. In our study, there was no control over that, so that can lead to a lot of variability. And one of the things we looked at was, okay, if we normalize the long-chain polyunsaturated fat acids to the total phosphatidylcholine pool, that could normalize some of the changes. So the reason we were interested in this was that so long-chain polyunsaturated fatty acids have been shown to decrease in plasma of AD brains compared to cognitively normal people. Like I said, we normalize the phosphatidylcholines, and what you see is that while the total amount of phosphatidylcholines doesn't change, it's not actually shown here, but we can see an increase in the long-chain polyunsaturated acid pool in treated versus normal. So again, you see a change, a decrease in these in AD and a reversal of that in the treated patients. Another category that we looked at was carnitines and acylcarnitines. So carnitines have an important role in acetylcholine synthesis. They're a critical role in cholinergic neurotransmission. They've been shown carnitine and methionine, which is part of the carnitine cycle, are significantly reduced in AD patients' serum compared to controls. And then, going back 20, 25 years, there are actually a couple of clinical trials where, in an effort to try and boost cholinergic transmission, acetyl L-carnitine was tested in a clinical trial. So there was one study, it was a year-long study, and they saw a benefit in a subset of mild AD patients. And then there was a follow-up study where in early onset patients that completed the trial, they saw some benefit there. So nothing further has been done with that, but it was interesting. So in our samples, what we saw in the placebo patients, we saw a decrease over time in the this is the acetylcarnitine. So a decrease in the placebo patients, but in the drug-treated patients, we didn't see any difference, and that was statistically different between those two groups. So, in summary, CT1812 is a Sigma-2 PGRMC1 antagonist, which displaces A-beta oligomers from the synaptic binding sites. We've shown that the proteins are in synaptic spines in cultured neurons where the A-beta binds and where the drug can act to displace the A-beta oligomers. We've seen evidence in plasma for mild to moderate AD patients that CT1812 can show a pattern of metabolic changes that oppose the direction that these metabolites have during AD disease progression. And we think that this small trial warrants further examination in a larger cohort and future studies that are set to commence, where we will be taking samples for analysis. So I just want to acknowledge a lot of people that have helped us along, people in our lab, a lot of whom are here today, our medical and scientific advisory boards, our academic collaborators, many of whom I've named, and of course we've been the recipient of some very nice funding from the National Institute of Aging and the Alzheimer's Drug Discovery Foundation, and of course the patients and their families who are taking part in our studies. So I just want to, again, one more time, we have some posters in the Sunday afternoon session. We hope to see you then and talk a little bit more. Thank you.
Maybe we should just go on to Susan and then ask questions afterwards. We're running behind.
Okay.
Like 10 minutes behind. I think you made me in charge of keeping people on time. You're doing a horrible job here, so try to get us back on track.
That's a great idea, Bob. Yes, we're running behind, but I will not be talking as long as my slots. We will catch up. So I don't think I need to introduce myself. Maybe Bob can introduce me from over there in the seat, but I think you all know me, so why don't we just keep going here? So starting with the same thank you slide, and really got to say, the team at Cognition is just absolutely stupendous. These are the people who did all of this work, and they're all here. They all deserve a round of applause because I just send emails for a living, so, that's how it goes. I just wanted to let you know we've had the great fortune of receiving quite a lot of support from NIA, both for the preclinical studies and the clinical studies. So this is really important to acknowledge the significant role that NIH has played in the absence, or I should say in the presence of quite a lot of clinical trial failures in this sector. Without them, there really wouldn't be much going on. So I just wanted to remind folks the rationale for targeting A-beta, because this sometimes gets lost and the baby gets thrown out with the bathwater in a lot of the discussion and debate that happens these days. So there really is overwhelming genetic evidence of A-beta's role in Alzheimer's pathology, not only from the early onset mutations where more than 200 of them really actually impact the concentration or the assembly rate or the ratio of A-beta protein, but also from the late onset risk genes, the biggest one of which is ApoE, that is the main clearance pathway for AD. We think that the reason why previous therapeutics have really failed is that A-beta can exist in a variety of structurally distinct forms, and Aladdin touched on this, but I thought it would be good to go into a little bit more detail here. What you're seeing is the structural equilibrium that actually has a Y shape in between fibrils on the one hand and monomer, which is an intrinsically disordered proteins, and oligomers, which are literally technically a metastable intermediate state. And so when you look at the therapeutic history of what folks have done in Alzheimer's disease, they've sort of tried to turn off the tap by inhibiting the two enzymes that cut A-beta protein out of the precursor. And so this would be gamma or beta secretase inhibitors. And so they do, in fact, effectively inhibit monomer production. This has been shown in very many studies, so they're very effective at what they do. But they had no effect on disease progression. And in fact, folks got cognitively worse, which was really a shame. For many of these inhibitors, the fibrils certainly remain in the brain as a source of monomer. So the fibrils can dissociate back to monomer and then go on to form more oligomers, especially at locally high concentrations you see around synapses. So on the other side of things, folks have tried monoclonal antibodies. And so these are antibodies that are directed against A-beta. And they did, especially in the case of aducanumab, a really effective job at clearing fibrils and plaques out of the brain. Some of those folks had fibril and plaque load that was reduced below the level of detection with amyloid PET agents. So it was really quite remarkable, but there was no effect on disease progression. And we think it's because those antibodies don't really engage with the oligomer target. So they're really not effectively dealing with the toxin that's causing disease. So Nick described to you our drug. It sort of sits in the middle of this equilibrium, and we've demonstrated in a variety of ways that 1812 can actually selectively displace that oligomer out of that binding pocket without having any effect on the monomer concentration. And that's key. So you really don't want to touch that monomer, and you really don't want to perturb what's going on in its normal physiological role, even though it's not very well characterized. We know that it's secreted in an activity-dependent manner at the synapse, and that usually means that it's doing something pretty important. So we and other folks have demonstrated that that oligomeric form binds to a single saturable site at synapses, that this causes the toxicity at picomolar concentrations in Alzheimer's brain. And what we identified through this phenotypic approach was an intervention point. So the point where you can stop the downstream toxicity that flows from it. And I won't go into this, nor will I spend a lot of time on this slide either, since Nick did a very effective job at guiding you through the preclinical evidence that suggests that all of that's true. So we went from our preclinical studies to first-in-man studies. This was a pretty routine single ascending, multiple ascending dose study in healthy young and elderly volunteers. And we saw that we had safety and tolerability at doses that were expected to produce therapeutic concentrations. This had been defined from the preclinical animal model studies as something that exceeded 80% receptor occupancy at the receptor in the brain. And so we then went on from there to our first in-patient study. And Nick showed you some of the data from that. This was a very short study in which we saw a lot of changes, including a reduction in synaptic damage markers. And you've seen some of this previously, so I'll go through this fast. Again, this is the study design, very short, 28 days. So this was the first patient exposure. And in 19 patients that were randomized to one of 3 doses of drug or placebo that was given by mouth once a day for 28 days. So I'm going to be showing you data from the spinal taps, so the protein expression in the spinal taps that were done at baseline and at day 28 of the study. So the first thing that we did was look for oligomer concentrations in the CSF. And so you remember Nick had taken you through that preclinical with the red dots, the graph all the way over on the right side. After a single dose of drug, we see an increase in oligomer concentration that's dose-dependent out in the CSF. And so this was pretty unexpected that we would see the drug not only displace it from the receptor site, but somehow facilitate clearance out of the brain, either directly or indirectly. We still don't know which is the case. So we went ahead and looked into patients, and the results from the trial are shown in the middle. On the y-axis, you're looking at the change from the patient's own baseline, between baseline and day 28. And the placebo in the diamonds and the hex is the pooled treated patients. And so what we saw was a significant increase in the concentration of the oligomers in the CSF compared to the patient's own baseline. We didn't see that in the placebo. The difference between the two groups was significant. And so this was consistent with the drug's mechanism of action, consistent with what we had seen in preclinical studies, and we thought encouraging evidence of target engagement. The drug had gotten into the brain and engaged the target. I've also spoken to you in the past about the protein expression changes that we saw out in the CSF as well as the plasma. Quite a number of proteins that are changing in pathways that are known to be impacted by Alzheimer's disease. We also took a look for specific proteins, targeting those proteins that are markers of synaptic degeneration. So this is an effort in Alzheimer's disease to really understand different classes of markers and what it's telling you about the degeneration that's going on in the brain. And so in the box on the left-hand side is a figure from the literature. Quite a number of folks have looked at CSF expression of fragments of synaptic proteins. In this case, it's neurogranin. This is a postsynaptic protein that's critical for synaptic plasticity. You can see it in the neurons in the dish in our preclinical studies on the right-hand side. It's normally really robustly expressed not only in the dendrites but also out in the spines. When you add oligomers, it just wipes out expression. But when you get rid of those oligomers by displacing them with the compound, the expression just is restored back to normal. So we knew that we wanted to take a look at this, and we did, and the results of the trial are shown in the middle. This was an analysis that was performed by Kai Blenoff at the University of Gothenburg. And so what you can see, same kind of graph in the middle there, change from the patient's own baseline in the ELISAs that Kai did with the change in the neurogranin fragment concentration increasing in the placebo patients and decreasing in the compound-treated patients. So this was not the only synaptic protein that had this pattern. We also saw something similar for synaptotagmin. Again, the same kind of pattern. In the literature, we know that fragments of synaptotagmin go up as the disease progresses, and we saw restoration of synaptotagmin expression in brain cells once you get rid of the oligomers. The results of the trial are in the middle, the same pattern. The drug-treated patients have a decrease in their CSF concentrations of synaptotagmin fragments compared to their own baseline, whereas the placebo patients increases, and the difference is significant. So lastly, we wanted to look at tau, and so this is new data since the last time we spoke to you. Tau is obviously hyperphosphorylated tau is the second major histopathological correlate of Alzheimer's disease. The neurofibrillary tangles are composed of tau molecules that are absolutely loaded at dozens and dozens of sites with tau. And so we know from the preclinical literature that the story is interesting, and it's starting to get more interesting as time goes on. So we know that when you put A-beta oligomers on neurons in the dish, you see an upregulation of tau kinases, and you start to see phosphate loaded onto the tau, and tau mislocalizing out of the axon into the dendrite, where it participates directly in synaptotoxicity. So we went to a colleague, Ian Pike at Proteome Sciences, who has a tandem mass spec method that allows him to look at where the phosphate is along the 400+ amino acid length of tau. So folks are starting to look at this. Where is the phosphate in that sequence? How many of them are phosphorylated? Because we think that the tau biology is going to be considerably more complex than simply what you would see at the end stage with the neurofibrillary tangles. So Ian has a method to look using tandem mass spec at both unphosphorylated and phosphorylated protein and reliably quantify 35, 36 of the known 39 sites where phosphate can sit on tau. And what they published, this is a study that they published in 2017, and here what you're looking at is a ratio between the Alzheimer's patients and the age-matched control patients, and the height of the bar indicates the degree of phosphorylation. And as you would expect, you see an increase at 11 out of those 35 phosphorylation sites. That's increasing by at least 30% above the noise level, which is the noise level, in the 80 patients compared to controls. Okay, great. This is as expected. This tells you where it is along the length of the tau molecule. So we went to Ian and we asked him to do the same thing in the CSF samples from the patients in our trial. And so the results are in the next slide. Again, on the left-hand side in red is that same figure I just showed you, the Russell paper. And the same lab, same method, same machine, same instrumentation. These are the CSF samples from our trial. And so here, what we're looking at is the ratio of the drug-treated compared to the placebo-treated patients. So you're looking at, as Tony likes to say, a delta-delta. So this is the change from the patient's own baseline, and now we're looking at the median for the two groups expressed as a ratio. It's a little bit complex, but it's meant to reflect and allow you to easily compare to the Alzheimer's patient graph, the published graph. So what you can see is that 6 out of those 33 phosphorylation sites decreased by at least 30% in the drug-treated patients compared to the placebo-treated patients. So that's a pretty broad reduction at a number of amino acid sites in the phosphates on the tau molecule that winds up in the CSF. So there's really only one way that that can happen, and that's if the drug is acting upstream of the kinases, those 5 or 6 kinases that are loading phosphate onto tau. So this is the first clinical evidence of a broad reduction in phosphorylation at a number of amino acid sites at tau, and it supports the A-beta oligomer hypothesis of Alzheimer's disease. It suggests that it's the A-beta and the A-beta in the oligomeric form that's setting in motion the changes that lead to what we see going on with tau. It certainly is consistent with observed reduction of synaptic damage, so that abnormally phosphorylated synaptic tau, which is something that Tara Spires-Jones' lab focuses on and studies quite extensively, which itself is directly synaptotoxic, is potentially being reduced by the compound. So I'll go a little bit more rapidly. So evidence, encouraging evidence for target engagement. Nick showed you evidence for disease modification, and we think encouraging evidence that there is synaptic protection after only 4 weeks of treatment with the compound, because we see a reduction in these damaged proteins as well as tau phosphorylation itself. So we've got a number of clinical trials going on right now under the able direction of our brand new Chief Medical Officer, Tony Caggiano. Wave, Tony, so everyone can see you. I didn't introduce you before, but there he is. We're very fortunate Tony joined us. And so those trials are looking at, I'll show you the clinical trial design in a second. Those trials are looking at various aspects of the kinetics with which the oligomers move out of the brain into the CSF. And we're also looking at, in more detail, at synapse number. And are conducting some really innovative clinical science with all of these, as well as a large efficacy study. So really briefly, I'll take you through a couple of them. This is the study where we're going to be measuring CSF fluid hour by hour after a single dose of drug, and we're looking for the kinetic response of those oligomers. We're looking to measure the oligomer response over time in the CSF of patients. This is a placebo-controlled trial, like all of the trials we've done, in which we're looking at either a single dose of drug or placebo to compare. So we're pretty excited about this one and what it can tell us about oligomer dynamics over time, rapid dynamics. We're also doing the world's first trial using the UCBJ imaging agent. This is a derivative of levetiracetam, and that's a drug that binds to SV2A proteins, an anti-epileptic drug. And the folks at Yale, Richard Carson and Chris Van Dyke, discovered that this is just an excellent PET tracer, excellent kinetics, and it can quantitatively image synaptic density in the brain of a living patient. So in this trial, which was originally 6 months and has now been extended for a further 6 months, what we're doing is looking at patients with mild to moderate Alzheimer's disease that have been treated with 1 of 2 doses of drug or placebo. And we're going to be doing an awful lot of imaging on these patients. We're going to look at synaptic density with UCBJ at baseline, 3 months, 6 months, and again at 12 months. We're going to be doing FDG PET at baseline and 6 months and 12 months. We're also going to be looking at resting state bold throughout the trial, as well as taking the cognitive tests throughout the trial. And we'll do the spinal taps at 0 and 6 months and hopefully get a lot of volunteers at 12 months so that we can look for the first time at changes in synaptic proteins in the CSF, changes in synaptic density centrally, and how both of those relate back to behavioral performance in the same patient over time. So we're really excited about this one. And so the efficacy study is the same 2 doses. We've got 2 cohorts here. Part A, I'm very happy to say, because of the just incredible results, incredible effort of the folks in this room, the last patient is going to get their final spinal tap from that Part A cohort of patients October 24th. So sometime in the new year, we should have results to report back to you guys about what we saw after once daily 6-month administration of Aleta for the first time. Same mild to moderate patients. We're focused on that throughout the clinical trials so far. Part B is just sort of scaling that up. Not as much imaging in this study, but we will be doing the cognitive testing and the spinal taps to look at the protein expression. And then finally, our brand new study, which has a brand new name, Sequel. So this is a supportive physiology study in which we're looking at the same thing we did initially, just a 1-month exposure with a washout and then a crossover. So this is a double-blind crossover study in which we're looking at the higher dose of 1812 and looking at its ability to impact theta power. So this is one of the EEG rhythms that's affected in Alzheimer's disease. So with that, I'll end hopefully early and take any questions you have.
So, very interesting presentations. Congratulations on the work to date of you and your team. I wanted to ask probably what is a very naive question, but is there any reason to believe that the compound Aleta has effects on its own, absent A beta? And the reason I say that to also suggest that or ask whether there's an appetite to measure microvessel blood flow. Just this year, Dave Atwell from the UK published that A beta actually acts in part by constricting the microvasculature, thereby resulting in diminished energy to the brain and causing all kinds of things. So I wonder whether, one, you might look at your compound on maybe some cerebral blood flow endpoints, but two, to ask the question, is the compound working only under conditions of amyloid overload, or does it in and of itself have any positive effects?
Sure. It's a very good question, Sonny. So we don't think that it interacts with anything else or on its own. And there's a couple of reasons for thinking that. First of all, the standard counter screening that we did at, I think, 100 receptors shows that we're very well separated 100-fold versus affinity at anything else. So that's not the universe of proteins, certainly. It always is a formal possibility, but we think this is truly selective for Sigma-2. Secondly, the way that we did the phenotypic screens initially, and that is what drove SAR, was to always test whether the compound was neuroactive on its own. So it was very important to us, since we knew that synaptic plasticity mechanisms are extremely sensitive and likely to be negatively impacted rather than positively impacted by any CNS active pharmacology, that we rule that out. We were deliberately looking for something that only affected the disease state and that did not have a nootropic effect, that did not augment normal learning and memory or anything like that. So as we all come to grips with Sigma-2 pharmacology and the state dependency there or the interactions of these pharmacology sites with each other, this is really interesting, but it is something that we deliberately took off the table at the very beginning.
I just want to add a comment to that. Some of the other activities that you've seen people talking about today, such as metabolic stimulation or caspase activation, that was part of our screening process too, and we selected, we have seen compounds that have those activities, definitely, and part of our screening was to select compounds that did not have those type of activities, but did have the anti-A beta activities.
Should definitely listen to Nick because he's the one who does the work.
Excellent talk. I wondered if you had a chance, I know there's a lot of data to sift through, but if you looked at all of those specific phospho-tau sites to see if they've already been cataloged, if you will, for how it affects the tau function?
This is an absolutely fascinating and emerging aspect of what we're all starting to look at in Alzheimer's disease. So the more we know about tau, the more we know that it's an extremely complex molecule. And what we had previously done through the history of Alzheimer's disease was sort of look one ELISA at a time. So one amino acid pair at a time, one amino acid site, one phosphorylation site at a time. And that's not given us a really good picture of this dynamic molecule and its potential roles, the potential roles of the phosphate at the different amino acid sites for triggering downstream biology. So I think the profession is just at the beginning stages of that. There's some work about to be published about using these kinds of tandem mass spec studies to look at the entire length and where the phosphate is along it. Obviously biofluid samples give you one perspective on that versus preclinical studies where you could really, get the full picture of what's going on. But it's a very active, important molecule and certainly can be directly synaptotoxic under some conditions. Okay. If there are no more questions, then... sorry. I'm sorry, Carmen.
What do you expect from the FDG study, the one that you will do with FDG? Because it is not the right, maybe, marker for CNS. So I don't know what you are going to expect with this PET.
Sure. So we included FDG PET. Certainly there's a long history of looking at this in Alzheimer's trials. There's a certain power associated with the hypointensity that's seen in Alzheimer's patients. It's a very dynamic readout of quite a number of things, glucose utilization, blood flow, oxygenation, a variety of things. But it was the canonical marker of synaptic function in the brain. And so comparing that directly to SV2A, this new synaptic density marker, was one of the rationales for taking a look at that in conjunction with the SV2A PET. Okay, so if there's no more no more questions, I'd like to introduce Mike Cahill. Mike is going to be attending via stream.
He provided a pre-recorded video.
A pre-recorded video, and then we'll switch over to questions from Mike live. So he'll be here in person to answer your questions. And I do want to mention the really impressive work and thought leadership that Michael has exhibited over the years with respect to this protein. So we're really quite interested in that. He is interested in moving back to the States. So I would ask you all to consider how we can bring Mike back here and have him be part of the community in the States here. So take it away, Mike.
Hello, welcome from Australia. Sorry that I couldn't be here, and it's a great pleasure to be invited nevertheless. Thank you very much. I'll be talking about PGRMC1 and its role in interest in biology. So here's just a schematic of the protein. Just draw your attention to those two tyrosides, which are phosphorylated. It has a cytochrome B5 fold. It is not related to the progesterone receptor. It's the archetypical member of the membrane-associated progesterone receptor family, or MAPR family, and these have a MAPR interhelical insert region between helices 3 and 4 of the canonical cytochrome B5 that defines the family, and they have also unique tyrosinate heme chelation, and that means that when the heme is reduced, it accepts an electron, it loses affinity for the protein, which leaves sort of a pocket there to do some other interesting stuff. So we have 4 MAPR subfamily genes that are there. Okay, I was first interested in this protein when back in Germany in 2008, we found that it was differentially phosphorylated between estrogen receptor positive and negative tumours, and that in vitro phosphorylation mutants affected cell viability. We also found, and we can see here, that the estrogen receptor is in an estrogen receptor positive tumour, that the estrogen receptor is downregulated in the hypoxic zone, and PGRMC1 is upregulated there. And this is the necrotic core of the tumour here. So, interestingly, in an adjacent slide, we see that PGRMC1 induction precedes the HIF1A induced hypoxia-induced factor 1A GLUT1 expression glucose transporter. Okay, so we then predicted that PGRMC1 was involved in the Warburg effect. Unfortunately, the Australian grant system didn't believe that that could be. They didn't give me any money, but since then, SABA has basically just confirmed this result in a very nice paper. You should read that. And we have similar results, largely on the back of Bashar Dajir, who I'll talk about those. So essentially, we have a system where we've mutated these 2 serines, which we think are negatively regulatory serines, and in the context of that, we then also mutate this tyrosine. So the idea is the 2 serines will open up the tyrosine for interaction, but once we destroy that, then it goes again. The wild-type protein doesn't change the shape of the parental cells. You can see the sort of fibroblasty-looking things. The double mutant, the 2 serines have gone double mutant. It changes the shape to this rounded shape, and the triple mutant also is a rounded shape, if anything more compact. We can see there we've got the Western blot characterization. The model that I'm looking at is 3 independent stable cell lines for each plasmid. So any differences we see here are not due to cell artifacts, but they're due to the protein themselves. So here we can see a proteomics analysis. There's the heat map at the top and the pathways map there. And basically these red pathways here sort of look like energy metabolism, and these blue pathways at the bottom look like ribosome translation, messenger RNA processing, some chaperone stuff, and things like that. I just draw your attention to 2 of those pathways, actin cytoskeleton and PI3 kinase, just to show you what's going on. So this is the actin cytoskeleton, half a dozen proteins involved in this, and actually when we try to verify this with scratch migration assays, we see that double mutant cells indeed have higher activity. the PI3 kinase activity, you can see that proteomics is predicting it to be down in triple mutant. And when we look at BAD or glycogen synthase kinase 3 phosphorylation, both of those are reduced in triple mutant relative to double mutant. So once again, the only difference between those is the oxygen of that tyrosine there. And so we can say that PI3 kinase ACT requires tyrosine 180. With glucose metabolism, both glucose uptake and lactate secretion follow similar profiles. Introduction of the PGRMC1 reduces that. The double mutant consumes much less oxygen, much less glucose, and the results are reproducible. So a very interesting effect on mitochondria. Here we can see in Mia, Packer and wild type cells that we have this long filamentous type mitochondria, and the rounded cells have these short fragmented roundy mitochondria. And yeah, you can read about that elsewhere. We show here that the tyrosine 180 is absolutely required for efficient mouse xenograft subcutaneous tumour formation. So we also involves tyrosine 180 single mutant there. So the effects on mitochondria prompted me to examine whether PGRMC1 may have been involved in the origin of mitochondria or the origin of eukaryotes. And the system that we're looking at there is at 2.4 billion years ago. Photosynthesis had just evolved, it's bacterial photosynthesis, and the methane atmosphere was converted to carbon dioxide and oxygen, which was not blue sky smiling at me, it was very life-threatening, and anaerobic proteoeukaryotes, which are an Asgard archaeal cell, needed a survival strategy, and that was the recruitment of a bacterial symbiote to detoxify its oxygen. That's a great paper, you can read about that there. So that study showed that bacteria, there are these cytochrome B5MY MAPR-like proteins that came from bacteria. They have no meheres, so this mehere is a eukaryotic invention, but they do have tyrosinate heme-chylation. So also the system has, all of the genes here came from bacteria. That is the mevalonate pathway, squalene cyclase, which produces the first sterol lanosterol, and the cytochrome P51 and PGRMC1, which then modify the first sterol lanosterol into this FF-MAS, which is actually a meiosis-inducing factor. So that's the possibility that PGRMC1 is involved in a meiosis induction. The reaction of cytochrome P51 here and PGRMC1 actually interesting because it involves oxygen. So you can imagine that when oxygen's around, this sterol is produced and it does something to save the cells from dying of oxygen stress. The genomic context of bacteria that have this cytochrome B5MY type protein, it's an inducible system, it has a ferric reductase and it has two other cytochrome B5. So it's some sort of a redox sensitive system that's induced. One could hazard a guess it's induced in the presence of oxygen. A model how that might work is that this the cytochrome P450 and MAPR protein make lanosterol. PGRMC1 loses its heme. The sterol then binds to PGRMC1, and somehow that increases mitochondrial activity and reduces oxygen levels so the host can survive. This also could be involved in other stuff, like for instance PGRMC1 is involved in mitosis, G1 checkpoint, membrane trafficking, and perhaps even the origin of the eukaryotic membrane. So what eukaryotes can do that other kingdoms can't do is pinch vesicles off to the inside, and this involves actin, myosin, cytoskeleton, so you can read about that in that review there. But PGRMC1's membrane trafficking could be related to this, and we see that we've got this motif of PGRMC1 here lined up against lots of myosins, called myosin actin, and in the myosin we see that the protein is that motif is in the coiled region. There's actually a slight break in the coiled here, and this is the sort of place where proteins are expected to be able to interact with those loose amino acids there. And in PGRMC1, you've got this short coiled and then a break, and so potentially myosins and PGRMC1 could be interacting with the same thing. It sort of reeks like, smells like cytoskeleton. The tyrosine is actually, tyrosine 139 is actually at the position of a heptad repeat, And so if this was phosphorylated, it would break coiled-coiled interactions and then would recruit other SH2 domain-containing proteins in there. So this eukaryotic invention of the me here may be involved in membrane trafficking. So we have proposed ancient PGRMC1 functions involving steroidogenesis, tyrosinate heme chelation, regulating oxygen consumption and metabolism, and maybe also membrane trafficking is related to the origin of eukaryotic membrane. So we also had a look at when the phosphorylated residues of PGRMC1 appear. This is lastly Elizabeth and Mikhail. If we just focus on the cytochrome B5 domain, we've got the choanoflagellate, single-cell sister group of animals, primitive animals here, and then we've got the placozoans. So the C-terminus of PGRMC1 gets bigger. In the cnidarians, it gets bigger, and the bilaterians, so the common ancestor of these has got an enlarged PGRMC1 C-terminus. It also inherited tyrosine 139 and tyrosine 180, and they've stayed in all animals subsequent. So those organisms, the common ancestor of bilateria and cnidaria, which I'm calling the K-back, it had the gastrulation organiser. We can see here Wnt-driven signalling leads to differentiation, in this case of sort of stem cells and tentacle-like cells, and in the Spearman organiser, it's lays down the body plan and leads to the subsequent differentiation of all those cells via a system of CpG methylation, among others. So if we just look what's going on here, this is the animal evolution. So actually hypoxia-inducible factor here was acquired by the placozoans. So all of these animals up here have that. Then the organiser was acquired here, and all of these animals have this. So placozoan don't have an organiser, but all the other ones do. And just then once again, notice that the placozoa is closer to the cnidarians than other ones. So the organiser then defines a nervous system, has synapses to striated muscle and localised coordinating ganglia, and is this related to oxygen tension and PGRMC1, tyrosine 139 and 181. When did all this happen? Here is the Ediacaran stratum, which is the first animal fossils. We can see that already we've got annelids, chordates, and some of the major groups already appeared by here. So between the origin of the K-back and this, then all of that's gone on. And the different molecular studies sort of say it's around here. The first animal was around here, and this is sort of beyond 700 million years. And this grey box here is what's called the Sturtian glaciation. It was the mother of all snowball events on Earth, 70 million years of frozen planet, and very low oxygen and low carbon productivity. So the origin of animals was associated with an oxygen stress level. This is somewhat reflected in modern embryology, where we have these glia cells expanding in the embryo, and they're driven by HIF1. When oxygen comes along and destroys HIF1, it then leads to the differentiation of neurons, and embryological HIF1 response is very uncharacteristically independent of Wnt beta-catenin signaling. So Wnt and beta-catenin are associated with angiogenesis, not with early embryologists. So once those neurons develop, then we know that PGRMC1 is involved in axon guidance and the embryonic formation of the central nerve cord. This is conserved from nematodes to mammals and involves interaction with deleted and colorectal carcinoma family. We also know that PGRMC1 is involved in active synapse function, and it's tyrosine phosphorylated only in synaptosomal fractions. So that leads us to a hypothesis where PGRMC1 function was affected in eukaryogenesis, and once again, to play with metabolism, it was affected at the time of the Sturtian glaciation, and this then led to the differential switch between metabolisms, which gave rise to the organiser. And the modern functional relevance of that is that PGRMC1 is potentially able to respond to hypoxia and or induce a hypoxic or pseudohypoxic state and revert cells to an undifferentiated state. So if we go back to more of Bashar's results, he's found that this N-nicotinamide methyltransferase, NNMT, is high up in triple mutant cells, and this enzyme catalyzes this reaction where S-adenosylmethionine methyl is transferred to nicotinamide, and this affects pools of SAM which are then not available to regulate genomic and histone methylation. So because of that, we took a tentative look at epigenetics. And I think this will be the single most, the strongest and most specific mechanism that we'll see today, that we have each of these PGRMC1 mutants takes its particular cell line to a very, very signature place, very far differentiated from other cells. So it clearly identifies PGRMC1 as a master epigenetic regulator. So just to look at one of those types of cell interactions there, we've got the triple mutant versus double mutant. So we're just taking the oxygen acceptor off that tyrosine, and we've got all these, hundreds of CpG sites become hypomethylated and thousands become hypermethylated. So if we have a look, then we can see that all of these in exons, unprimed translated body, exon boundaries, transcriptional start site, all of these are more methylated in triple mutant. It turns out triple mutant is the most methylated of any of these cells. So I was interested where the hypomethylated sites were, and actually it turns out that they're in cell type of promoter-associated enhancer. So in the context of global methylation, we have some cell and promoter-specific enhancers which are hypomethylated. And of course, this is just very reminiscent of induced pluripotent stem cells, where the level of methylation goes up and resembles those of the human embryonic stem cells. And that, of course, probably resembles the state of cells in the KBAC at the stage where this organiser eventuated. So if we look at the GO pathways, then all of this neurogeneration, animal organ development, organ morphogenesis, all of these pathways are saying this is what would happen if we played with something that was related to organised biology. So we know that pluripotent stem cell pluripotency is affected by one, the NNMT pathway, which globally regulates SAM levels, and two, by PGRMC1 via catenin and P53. Actually, the second paper doesn't quote the first one, but what we're providing is a mechanism which connects those. Interestingly, the stem cell system is one where PGRMC1, the cytochrome B5 domain, is extracellular, just as in the Alzheimer neurons. So in an Alzheimer's meeting, the relevance or in a neuroscience meeting, the relevance to Alzheimer's. This is something I presented last year based on Tim Kennedy's group, that netrin signaling in presynaptic activates postsynaptic DCC. Calcium signaling leads to a new AMPA receptor gene transcription. This then enables the cells to respond more positively to subsequent stimulation, to remodel the postsynaptic spine and lead to stronger synaptic connection and LTP. Notice that membrane is 40% cholesterol. So in Alzheimer's, we have this A-beta oligomer binds to the Sigma-2 receptor, which I've got somehow putting PGRMC1 in a different non-compliant form, and so all of that stuff doesn't happen. So PGRMC1 DCC is central in my model here. We've got here the cognition therapeutics drug is coming and affecting this model, and then allowing PGRMC1 to then do whatever it does again, and this leads to LTP. So what I would add to that now is that maybe we've got some sort of Warburg or pseudohypoxia effect going on here. If this is kind of not absolutely crazy, then we expect to find mitochondrial epigenetics and glucose metabolism defects in Alzheimer's, and we also, all the usual suspects, tau, amyloid, and ApoE should fit into the model somewhere. So we can look at mitochondria here, and we can say, yes, they're affected. Epigenetics, absolutely. Glycolysis, really important for neurons, and we can see glucose dysmetabolism precedes onset. So glucose, absolutely yes. The usual suspects, tau, A-beta, and ApoE. So it turns out that APO, as a component of a lipoprotein, is responsible for clearing cholesterol esters. And when we play with that system, you can read in this paper that we get reduced tau and A-beta pathology. And also from Riyad et al. Last year, that the ApoE is probably involved in PGRMC1 LDL receptor trafficking, and I'd also direct you to the talks by Riyad and Katina in today's program. So the story that we've got here is we've got this sort of differentiation going on. PGRMC1 is involved in embryological response. Somehow that's perturbed in Alzheimer's, according to the model. CT1812 then comes in and fixes that up and it restores LTP. So that's what we're talking about. So if PGRMC1 really is involved in all of this, then have the usual Alzheimer's suspects perhaps blinded us to alternatives, and PGRMC1 could be an omega molecule for Alzheimer's and other diseases. So having said all of that, I'll just leave you with this, that perhaps the question is, could the DCC interaction do this alone, or is freeing up PGRMC1 allowing it to do lots of other stuff in addition to interacting with DCC? So I'll leave you there. Those are my collaborators, and thank you very much for your time. I'm happy to answer questions. Bye.
So you can click join. Yeah, we'll just drag them up at the right.
Hello.
Hi, we can hear you.
Excellent.
Okay, questions for Mike?
Thank you, Mike. That was a great talk. I'm happy to hear HIF, PGRMC1, and Warburg all mentioned in the same talk. Could you elaborate a little bit on the relationship between HIF and PGRMC1? I believe you said that PGRMC1 precedes HIF expression, or could you clarify that?
This is an experiment from a long time ago, but, well, you saw the data, that's all we have. We looked at GLUT1 expression. So what we looked at was GLUT1 and PGRMC1. I can't go any further than that. I don't know any more than that. That experiment happened 12 years ago, something like that. So GLUT1 was induced after PGRMC1. Whether PGRMC1 is also HIF1 induced, I don't know.
Thank you.
I thought, by the way, that our talks were kind of looking through the mirror at alter egos of each other.
Other questions?
So Mike, this is Susan. I was wondering if you could just elaborate a little bit more on the link between the glycolysis and the Spearman organizer.
Oh, that's there's a whole other aspect of this that I didn't kind of have time to unpack there. But if we think that PGRMC1 came from eukaryogenesis, and basically it was a story about what will I eat? The cell's thinking, what will I eat? And if I eat this substrate, then I get protected from oxygen. If that's hardwired into the genome, maybe that's what's going on when the organisers do, because like an organism at this grade, it's only got epithelium and endoderm. There's two cell grades. And there's some oxygen thing going on there, and the ability to change between those may have given rise to the organizer to be able to switch a cell type on and off rather than have different functions at different places in the cell. To actually have two cells doing something else may have given the program that the organizer's built on, like a foundation brick. There's another interesting aspect to this is the gut. It's the first thing that forms in gastrulation, so it's also an organizer product. And if we look at the gut, say in the crypt cells of the epithelial cells, then you've got hypoxia in those stem cells down there. And then as they mature and differentiate into the villi epithelium, the colonocytes, then that becomes an oxygenated environment. And those cells, it's very important for those cells to be undergoing oxidative phosphorylation to keep the lumen of the gut hypoxic. So that there's a kind of an interplay going on between the colonocytes and it's Warburg metabolism in the crypts. It's happening. And this affects the microbiota in the gut, and that in turn affects how the gut signals to the brain. And absolutely, the microbiota is, for instance, involved in risk for Alzheimer's disease. So you've got things like GLP-1, PGRMC1 interacts with GLP-1 receptor, and GLP-1 is one of the things that's released by the gut and goes to the brain and the pancreas to induce insulin and things like that. But very big potential for crosstalk there, and all related around this hypoxia axis. So, fascinating area.
Thank you, Mike. Any further questions from anyone? Okay. All right, then we're going to move on to closing remarks by Bob, who is already at the podium. Are you all set there,
Thanks,
Bob?
Mike.
Thank you, Michael.
Thanks.
Okay. Guess, everyone hear me okay? All right. As Susan mentioned, I'm here to make some closing remarks, and as the person who makes the closing remarks, I guess my job is to make some, simultaneous, outrageous and thought-provoking comments for us to think about for the next year, so that we have things to work on when we have the 5th International Symposium on Sigma-2 receptors, and I certainly hope we do. And before I go into a summary of today's symposium, what I thought I would do is kind of take a walk back in time and explain why Susan and I have been historically the chairs of this symposium, how our collaboration came about, and why we started having these symposiums. And in my opinion, the importance of our having this symposium and why I believe it's important to do so going forward. I think Susan and I first had a conversation is when she read this paper that was published in Nature Communications in 2011, and Jinbin Xu in the front row is the first author on this. And this was a paper that was prompted by some cancer research I was doing, where we showed that the Sigma-2 receptor was a very good marker of cell proliferation, but for many, many years, for decades, I had faced the question as what is this protein, what is its role in cell proliferation? Because until you can tell us what the protein is and what its role in proliferation is, we don't believe a thing you're saying. Okay? So we went down this path where we did a series of photoaffinity labeling studies with this compound WC21. We tagged it, we purified it, we sequenced it, and we came up with this protein, PGRMC1. And when we published this, when we were debating publishing this paper, we knew that the data was going to be somewhat controversial, because we knew there wasn't a one-to-one relationship between protein levels and radioligand binding of things like DTG or we looked at actually the RHM series of compounds. But we knew the data was just too strong to show that there had to be an association between the Sigma-2 receptor and the PGRMC1. So we wrote this paper saying that we think this binding site is somewhere in this PGRMC1 protein complex. We knew it wasn't the PGRMC1. We knew it was somewhere, we thought it was somewhere within the complex. And after some debate, we decided to go ahead and publish this. And believe me, we really debated for a long time on whether we should go ahead and publish this paper. And then Susan contacted me, and she was started telling me about some of the work they were doing with some of their compounds and how they bound to the Sigma-2 receptor and has some very interesting properties in Alzheimer's models. So we started working together, and the interesting thing from this collaboration was this paper where the group in cognition, with a small part from studies in our lab, showed that when you either treat neurons with a PGRMC1 antibody or knock it down with siRNA, it essentially behaves the same as a Sigma-2 antagonist. That was actually followed up by a study by Steve Martin's lab in Texas, where they were able to basically show the same results in a nematode model of Alzheimer's disease. So we were very excited, because now we had two studies that kind of corroborated our Sigma-2 PGRMC1 story. And everything was right with the world until, I think, Carmen, your paper came out in 2015, was it? And Carmen, her famous non-identity paper where she did short hairpin RNA studies to have a permanently knocked down cell line show that when you do that, it doesn't affect the binding of Sigma-2 ligands, and that we need to really rethink what's going on here. At that time, Susan called me and said, you know, Bob, there's some controversy about this result. Maybe we should get together and have a mini symposium or a nano symposium at Neurosci and get the people together and kind of hash this out. In between that conversation and the meeting, Arnie Rojo published his paper, basically showing the same thing that Carmen reported, except this time using CRISPR techniques, and that is when you knock out PGRMC1, it really does not do anything to DTG binding, and Carmen mentioned that it also doesn't affect cytotoxicity of Sigma-2 binding. So we had this meeting, the first meeting, it was in Chicago 5 years ago, and I think for anyone who was at that meeting, they remembered how lively the discussion was regarding this controversy, and we left that meeting thinking that what we did, our paper in 2011 had some serious problems. That was followed up with another meeting, the second symposium, I believe in San Diego, where there were similar results. Carmen's group showed some fluorescent properties of the fluorescent ligands that did not change when you knock out PGRMC1. And then there was a talk from Japan, we were looking at the dimer, where they identified this very low affinity DTG binding site, but was totally unrelated to the Sigma-2 receptor. So after the second symposium, and again, I'm going to focus my talk on the protein and not the other talks of the symposium, because this is where the greatest controversy was. So after the second symposium, we went back still equally confused as to what was going on. When Susan contacted me and said, Bob, it's time to think about the third symposium, I said, no, the biology of this protein is just evolving too slowly. I'm not going through another one of these until we can kind of figure out what's going on here. So we took a year off, and I think it was very important we took that year off, because in that year off, that Steve Martin and Andrew Cruz published this now seminal paper where they actually identified the gene for the Sigma-2 receptor. And their data was very strong, showing that this was the gene, this was the protein that was responsible for what we are calling the Sigma-2 receptor. Now, many people applauded this because they thought this was the final nail in that the coffin in that 2011 paper saying, this shows that this is wrong. But that's not the way we looked at it. We went back and we looked at the information at this paper. We looked at some information that was available in Mike Cahill's seminal review articles on the PGRMC1, and we looked at other data that was in my lab. I said, wait a minute, there's something more complicated here. We think that our conclusions are correct, and that this study from Cruz actually provided a very key missing piece of information to pull it all together. And as the lab reported last year, and as we published in scientific reports, we know that our initial conclusions were largely correct, that the Sigma-2 receptor binding site is located in the PGRMC1 protein complex, that protein complex consists of the LDL receptor, the TMEM97/Sigma-2 receptor, and PGRMC1. And if you take the conclusions of our paper and you substitute Sigma-2 receptor binding site with Sigma-2 receptor protein, you get the absolute right conclusion, okay? It's just we could not go that far because our data did not support that. But I think the results that we published initially in 2011 were scientifically valid, and I think this paper in Scientific Reports really substantiated that, and it showed exactly what that complex was. And what it is, and the second part about this paper is what's shown on this slide, is because for many years, people looked at the pharmacology of Sigma-2 receptors. I mean, you have a Sigma-2 receptor compound. It changes a lot of things within the cell, but we didn't know what it really did. This receptor was key because now it tied this protein to a very specific biological pathway, the internalization of LDL by the LDL receptor. The fact that we see that when you have the intact complex, the rate of the internalization is much faster than when you knock out either one of these proteins or both of these proteins. it is perfectly consistent with why this protein is upregulated in proliferating cells. That proliferating cells have a very high cholesterol demand. When cells are quiescent, the monomeric form of the LDL receptor is able to internalize enough. But since that demand increases, when cells start to proliferate and cycle, you form this complex and you increase the rate of receptor internalization. Okay? It makes perfect sense with what we were observing in cancer cells. A second paper talk we gave last year was by Chembo, and she subsequently published it in Cell Death Discovery. And one of the things we looked at was the binding of these various sigma ligands and the effect of knocking out the Sigma-2 receptor, TMEM97, and PGRPC1 in these cell lines on cytotoxicity. And we showed that RHM4 is very specific for Sigma-2 TMEM97. When you knock it out, there's no specific binding. When you do the double knockout or the single knockouts, you get a residual binding site for DTG, a binding site that has a KD of around 300 nanomolar. But the most important part of this talk is that we showed that was when you knock out either TMEM97, PGRMC1, or both of them together, it has absolutely no effect on the cytotoxic effects of Sigma-2 ligands that kill cells. Now, this was kind of a revelation, and it had me a little bit concerned because, you know, I'm probably as guilty as many people, and a lot more, or guiltier than most people, in putting out in the literature the use of Sigma-2 agonists as potential chemotherapeutics. And this paper was it was exciting to see, but somewhat kind of disappointing to see, because what that shows is everything I was saying was really not true. Because although they may be Sigma-2 compounds, but the cytotoxicity has absolutely nothing to do with the Sigma-2 receptor. And the important thing, and I think that what this answered, is there was always this big question in the field, is why do these compounds with nanomolar affinity, in order to be cytotoxic, have micromolar, 10, 20, 30, 40, 50, 100 micromolar concentrations to kill the cells? And the conclusion that Chembo stated in the paper is the reason why there's this disconnect between affinity and EC50 in the cytotoxicity assays is that because the cytotoxicity has nothing to do with the Sigma-2 receptor, hence nothing to do with this binding at the Sigma-2 receptor. And we think that this is associated with some secondary off-target binding site, the structure and identity of which we simply do not know. Okay, that was last year's talk. Let's go through today, the talks we heard today. I think the talks we heard today can be broken down into three areas. There's groups that focused on the TMEM97/Sigma-2 receptor biology, and it's look at it in understanding its role in Alzheimer's disease. We had a very nice talk from Dr. Colomb looking at the interaction of A-beta with the TMEM97 using his very high-resolution microscopy techniques, which we think that kind of is a follow-on study to what cognition did with the PGRMC1 years back. We heard a talk from Halad and Riyad, who showed that complex, that trimeric complex, is responsible for the internalization of A-beta, whether it's in the oligomeric or monomeric form. And he showed a very strong correlation with apolipoproteins, and I'm going to come back to that in a minute. And we heard a very exciting update on the studies being done by Cognition's compound CT1812. And Susan, I think the data that you and Nick presented is extremely important. It's extremely compelling. The fact that you're measuring these changes in the CSF levels of important markers that are indicative that these compounds are going to protect neurons is very exciting. The world is waiting to hear how does it work out in your cognitive performance. Does it actually slow the loss of memory function in people who are likely to have Alzheimer's disease? Also very interested in hearing what the results of your Yale study that show the preservation of synapses. So I think the series of studies that are on the role of this receptor in Alzheimer's disease are generating some very exciting results, and something to keep an eye on going forward. We heard three talks on different aspects of Sigma-2 receptor pharmacology and biology. Wayne talked about the metabolic stimulation of the cancer cells. Jinbin talked about the autoradiography of Sigma-1 and Sigma-2 receptors in postmortem brain, including Alzheimer's brain. I think it would be nice, Jinbin, you had a lot of scatter in some of your data. It'd be nice if you can go back and actually look and see if there are any sex or gender-based differences in the density of these receptors and the ratio of those receptors. Because I think there is based on other discussions we've had today, there's likely to be something there. And of course, Carmen presented her talks on trying to separate the cytotoxic effects of Sigma-2 ligands from non-cytotoxic effects of Sigma-2 ligands. And it's nice to see that those rigid analogs of the RHM series of compounds don't really have any cytotoxicity, because that shows you that the SAR of the RHM compounds is preserved in your compounds. Okay? And the third series of talks are looking at PGRMC1. We heard Sunny gave a talk on this microRNA LAT7I, and he looked at the effects of PGRMC1 and the release of BDNF in stroke models, and he made the comment that it looks like the biology of the PGRMC1 in stroke may be a little bit different from Alzheimer's disease, and I think that kind of makes sense, because one of the things that the cell biologists have always told me about is that to understand protein function, it's tissue specific and context dependent, right? And what you have are two very different contexts. One can imagine that the function of those receptors are different under those two different cases. In a talk that only Mike Cahill can give, he gave us a lecture on the evolutionary biology of the PGRMC1, and to show the complex biology associated with that and the different effects that these various point mutations have on different biological pathways. So now what I'd like to do is return to a slide that Susan showed earlier, and I told you I was going to change it, and I did change it. And I think the picture is starting to emerge that is consistent with what we see on this slide. And that is, I think we have a series of observations and results on the Sigma-2 receptor that are specifically related to and associated purely with TMEM97. And those are shown in this region of the slide here, okay? I think we have now, I changed the color because it's gray, because this is a gray area, because we now have a number of pharmacological effects that we are seeing with Sigma-2 ligands, but we don't know if they're really due to these ligands interacting with the Sigma-2 receptor. For example, the cytotoxicity effects, the caspase activation effects. I showed you that has nothing to do with the Sigma-2 receptor. At least I don't think it does, based on the paper that Chembo showed. Are some of these other pharmacological effects, do they fall into that category, or are they really related to Sigma-2 TMEM97? That's where I think we're really waiting to hear your results in the TME knockout cells. If you would like to work with any of the cells that we have generated in my lab, I would be happy to provide them to you, because I think that will save you time, and I think the results of those experiments are extremely important in moving the field forward. So Wayne, I'm offering whatever help we can for you to do those experiments. And then we have another area here, and I think this is maybe related to what Sunny and Mike talked about, because the biology of PGRMC1 is very, very complex. And I think that there is a series of there is biology associated with TMEM97. But one of the things Aladdin pointed out to me when he was doing his microscopy studies and his PLA studies is that there's a lot of PGRMC1 that's unassociated with TMEM97. And given the diverse biological pathways PGRMC1 is associated with, it stands to reason that there are other complexes in the cell membrane that the PGRMC1 can form complexes with that are not related to TMEM97. And that's maybe what some of the things that Sonny, you're looking at, and Mike Cahill has reported. But PGRMC1 is a very, very complicated protein. I think the thing that stood out in my mind is, I think when some of these Mike mentioned, is that actually some of these things at PGRMC1 actually may be downstream from TMEM91 PGRMC1 complex in that internalization process, where it then kicks around and hits another aspect of PGRMC1 biology. So it's certainly something that can happen. the biology is so unclear at this point in time, it really is going to take a huge effort in the field to try to sort through all of this stuff because it is a very, very complex biology. I'm going to come back to a slide that Aladdin showed because I think this was an important point that because he had so much data to present that he kind of went over quickly, so I want to make sure that we cover this. And in his data where he looked at the oligomers alone and the oligomers bound to ApoE2, E3, and E4, he found this rank order potency that is, I think, striking. He found that the ApoE, the oligomers bound to ApoE, had a lower uptake than the oligomers itself, and that ApoE3 was greater than the oligomers bound to ApoE3 was greater than the oligomers alone, but every bit important, the ApoE4 was very high. Why is this important? We know that ApoE2 actually is protective, right, Susan, in Alzheimer's disease, whereas the #1 risk factor in Alzheimer's disease is ApoE4. So we now have a receptor associated with the toxic species of A-beta, right, the oligomers, that is associated with known biology. It was associated with risk factors and neuroprotective factors. And to my knowledge, this is the only protein of all the so-called A-beta oligomer binding sites that has really linked with the clinical neurobiology of Alzheimer's disease. So either that is a huge coincidence or there's something there, okay?